Next, I would like to welcome Mr. Rudra Banu Satpathy, the founder and head of Technorate Groups. Next, I would like to welcome our conference chair, Anil Philip, Associate Dean, School of Pharmacy at University of Nizwa, Sultanate of Oman. Next, I would like to welcome Mr. Sid Charger, Managing Director from Technorate Group. Welcome all. Thank you for joining. Next, I would like to welcome our eminent keynote speakers. We are being joined at Dubai by Dr. Razia Khanum. Welcome, ma'am, professor and lead pharmacology from Gulf Medical University, Ajman, United Arab Emirates. Next, I would like to welcome Mohammed Haitham Ayed, R&D Manager and Fellow from Johnson & Johnson, Dubai. Welcome, sir. Next, I would like to welcome Dr. Ibrahim El Sayed, Director of Quality Assurance and Institutional Effectiveness from Gulf Medical University, United Arab Emirates, and Faculty of Pharmacy. Next, I would like to welcome Professor Marcel Muzaffari, the President and Founder, Australian Nanoscience and Nanotechnology Initiative, Australia. Welcome, sir. Next, I would like to welcome Dr. Ranendra N. Saha, the Director, and Shri B.K. Birla and Srimati Sarla Birla, Chair Professor, BITS Pilani, Dubai, United Arab Emirates. Next, I would like to welcome Dr. S. Gasmi, the Assistant Professor of Cellular and Molecular Research Center from Sharq University of Medical Science, Iran. Thank you all for joining. Next, I would like to welcome and introduce to our session chairs of the day, Dr. Jayavardhan Patil, Dean from Department of Pharmacy from Sankal Chand Patel University from India, Dr. Professor Nahla Ismail, the Council Member from Malaysian Academy of Pharmacy, Malaysia, Nida Bawarsad, the Assistant Professor of Pharmaceutics from School of Pharmacy from Iran, Dr. Bunta Venkata, Senior Lecturer from University of Malaya, Malaysia, Dr. Firoj A. Tamboli, Head and Department of Pharmacognosy from Bharati Vidyapeeth College of Pharmacy, Maharashtra from India, Dr. Azimova, Senior Lecturer from Turkey. Thank you all. Next, I would like to request Mr. Siddharth Charger, the Managing Director from Technorate Group, to address the gathering. We are sorry that the audio from Dubai is not audible to us, but it will be connected soon.
Now it's audible, Cecilia? So APR is built from humanity, so we stick to our commitment of delivering uninterrupted academic and scientific services to pharma professionals all across the globe in spite of challenges and circumstances. Amid the pandemic, we opted the online platform and delivered more than 50 webinars and couple of grand conferences, thus creating immense impact of research opportunity towards drug discovery. Today, APR is no more an association alone. We are a conglomeration of more than 100 international universities standing with us, making us one of the largest populated associations with more than 7,000 professional and student members. We are rapidly expanding our network and collaborating with universities, research centers, and industries all across the globe with a vision to establish world's strongest scientific network. The community will shortlist the qualitative research from grassroots to crest and divert with trial and implementing towards needs of mankind. Our mission is firm that our objective is strong enough to connect organizations and individuals of common research interest by creating a collaborative environment for knowledge sharing. Few golden words of His Highness Humaid Al Qatim, Honorable Director General of Dubai Health Authority, reverberated in my mind, who spoke about several areas where India and Dubai can collaborate. Moreover, I belong to the largest democracy in India, the nation which ranked 3rd and 14th in volume and values respective for pharmaceutical productivity. Dubai has been a center of globe logistically, thus providing a gateway to African nation to supply healthcare essential. I believe a collaborative environment between pharmaceutical academic and research organizations between both the nation will be add on global potential beyond the borders. I urge individuals and organizations to come front and be a flag bearer in respective field and geographical location to support toward mission of integrating research opportunity. I express my gratitude to all my colleagues, all members, scientific and national ambassadors, chair, speakers, and universities for their support to add on value of this conference. I greet everyone to share their experience, knowledge, and skill irrespective of nationality to make this conference furthermore productive and worth of attending. I wish everyone healthy life and happiness. May Almighty bless all of us, both of them. Thank you very much.
Thank you, sir. Thank you so much. Next, I would like to welcome our conference chair, Anil Philip, the Associate Dean from School of Pharmacy at University of Nizwa from Sultanate of Oman. Welcome, sir. Can you hear us?
Yeah, I can.
Thank you for joining, sir.
Yeah, thank you. I think there's a video issue with me. All right. It's okay if I don't put my video on.
Okay, no problem, sir.
Look like
yes.
Yeah, it's okay. Yeah. Thank you. Right. Very good morning to all the organizers and the participants, and I believe students also. As I see the agenda, there are a lot of students, and being from is the cornerstone, the pillars of any conferences. So welcome one and all, and I feel honored to give this address to all of you and all the participants of the 9th World Conference on Pharmaceutical Sciences and Drug Manufacturing. We all know that what pandemic has done to research in general, but the initiative once again by the Association of Pharmacy Research to conduct this conference will keep things back on track. Okay, and healthcare, as you all know, is going through one of its biggest shifts, and the advent of industrial revolution, IR4 as we commonly call it, has just added to that. And with modern technologies, I believe that the healthcare will be better and affordable. All we require is the active integration of these technologies and for the translation research. I believe one of the major aims of the conference, as I was going through, is to bring different stakeholders together in the pharmaceutical arena to share new results, new innovations in the field. As I look into the agenda with the different presentations that are there, I feel that there are varied innovators who are here, who are showcasing their research, their innovation. And I believe that a lot of innovative ideas will be shared within the next 2 days. And a lot of learning and networking will occur in this conference. This year's conference will, of course, have a lot of topics of varied from the varied disciplines, with the focus on pharmaceutical science and drug manufacturing. And I hope that this conference will motivate one and all to not only achieve their personal goals, but also it provides a forum for discussion and to discuss important topics in this field. Additionally, I hope the participants have a deeper understanding of the cutting edge research and technologies that are in this field, and which will be talked about or talked on by the leading experts in this field. I wish all the organizers and all the participants a great time and a safe and healthy conference. Thank you so much.
Thank you, sir. Thank you so much for your kind words. Thank you for joining. Next, I would like to welcome our publication chair, Dr. Aysu, Assistant Professor, Department of Pharmaceutical Technology, Faculty of Pharmacy from from Turkey. Dr. Aysu, can you hear us?
Hi, Elsa. I hear you.
Yeah. Thank you, ma'am, for joining.
Sure. Thank you very much again. Dear colleagues and friends, on behalf of organized committee, I'm honored and delighted to welcome you to 9th World Conference on Pharmaceutical Science and Drug Manufacturing. And this conference is the continuation of highly successful series of global meetings about pharmaceutical research. And as we know, Association of Pharmaceutical Research is renowned and specialized in managing pharmaceutical conference throughout the globe. It is the important things, discovery and development of drugs against human disease involve a wide range of scientific approaches of pharmaceutical science. Therefore, our keynote speakers will introduce and discuss cutting-edge developments in a large number of areas among novel drug delivery technology, including major challenges. Also, this conference will address the critical issues in the scope of drug targeting, nanopharmaceuticals, molecular modeling in drug designing with 3D printing drugs. And I can say, drug delivery systems applied to vaccine development. As we know how it is important in this COVID-19 pandemic. Also, our keynote speakers introduced the organs on chips, smart delivery system in pharmaceutical research, and including biopharmaceutical drug design, development, pharmaceutical chemistry, and research advancements. The intention of the conference is to emphasize the important role of all fields related to drug discovery and development process, and also provide a wide and unique platform for international collaboration and communications. Also, it shows us the communication opportunities in the interdisciplinary fields of pharmaceutical science by bringing the experts together from all over the world. I extremely hope you all find this conference highly engaging and beneficial for your future venture. This Congress, remarkably successful in all aspects of pharmaceutical research with all of us. And my last verse, and I'm sure this conference will meet your expectations and stimulate new collaborations. Once again, Yelisa, I am saying I thank you all very much and wish you all enjoyable and successful conference. Thank you very much.
Thank you so much, ma'am. Next, we have the release of the proceeding book. I request Dr. Razia Khanum and the keynote speaker, Mohammed Haitam Ayed, who have joined us at the venue, and our managing director from Technorate Group, Mr. Siddharth Charger, to kindly release the proceeding book of the year 2021 of the 9th World Conference on Pharmaceutical Science and Drug Manufacturing, to do the honors. Oh, book. Congratulations on the release of the conference proceeding book, and thank you, dignitaries, for doing the honors. Next, we'll be moving on to our keynote presentations. Firstly, I would like to welcome our keynote speaker for the day. Yes, I would like to welcome Mohammed Haitam Ayed, R&D Manager and Fellow from Johnson & Johnson, Dubai. Mohammed Haitam Ayed is passionate about innovation in drug delivery strategies which provide concrete benefits to patients. He is having more than 20 plus years working experience in pharmaceutical research and development, including strong expertise in formulation development of oral dosage forms, CMC, and drug delivery technologies, bioavailability enhancement, control release, and many more. He's a team leader and project management experience acquired in diversified industrial environments, big pharma, biotech, and contract manufacturing companies. Recognized expert in selection, audit, and management of CDMOs. And his specialties are formulation development of oral dosage forms, drug delivery technologies to handle specific drug substance problems such as poor solubility and short half-life, process development, scale up to industrial facilities, technology transfer, and troubleshooting. I request Dr. Mohammed Haitam Ayed to deliver his keynote presentation. Welcome, sir.
Hello, do you hear me? Is it fine now? Okay. Bismillah ar-Rahman ar-Rahim. I'm very happy to be among you today in this interesting and I hope very useful and successful conference. I would like to speak about a subject I am passionate about, which is open innovation in life sciences. So the presentation today, if you can move to the second slide, please. So the outcome of our presentation will be, we'll start by defining what is open innovation. We will talk about open innovation in life sciences, why we need it, what are the process of establishing open innovation, especially in collaboration between industry and academia, as I believe a lot of you are coming from academia and they are interested to understand what our pharmaceutical industry is looking for and what is the process for collaboration. And finally, we will conclude this presentation. Of course, being hybrid models, so there's a lot of difficulties. So hopefully you'll be able to solve it and move to the next slide, please. Yes. So basically, I would like to start, take a step back before talking about open innovation. Try to
hello. Voice is not good.
Is it better now?
Yes, sir. It's audible now.
Okay. Okay.
Thanks.
Okay, so basically, I would like to take a step back and try to define with you what is the difference between innovation and research and development. So basically, research is an activity, creative activity that creates knowledge. And this knowledge is what we call it, fundamental research. And this fundamental research is used in applications to have the applied research. While on the other hand, if you move to the next one, please. Innovation is the ability to transform this research to application to a product that are useful to patient, for example, drugs. So innovation is the capacity of commercializing research. So here in this diagram, you can see that we take the process start by having funding, okay, usually coming from public funding. And this funding is used in research and development activity to create knowledge. And then by using innovation models and approaches, we transfer this knowledge to products. For example, drugs can be sold, and part of this benefit can go back to research and development. So this is the cycle that we would like to establish in order to have all the elements. So basically, research and development transform the funding, the money on knowledge, while innovation transform knowledge to products, to drugs in our case, that can generate, again, the money for reinvesting. So I think this is very important part to be understood between the relationship between innovation and research and development. So if you move to the second slide, please. Of course, we cannot, talk about innovation and research and development without talking a little bit about our region. What is the status of these activities in our Arabic region? And this is, I think, true for all the Middle East area. So basically, our Arabic region having in my opinion, two difficulties when it comes to innovation and research and development. The first one is that we are not investing enough in research and development. We are producing, our region producing about 6% of the global GDP, but we are investing less than 1% of the global spending in research and development. And to make it worse, this small amount of investing in research and development is not going to produce products that are useful to the society, because most of it are carried, in academia and with objective to make publications. So here we have another difficulty. And another difficulty to transform this research to innovative products is the lack of infrastructure. Like, for example, life science incubators. We have very little life science incubator in our region, and we have very little fiscal incentive for industry to invest in research and development activities. For example, just to share with you, I mean, when I was managing a formulation development laboratory in France, at the end of each year, I used to submit all my expenses, the salary of my team, the equipment I used to buy, even publications and the patent that we used to file, all the cost, research and development costs, we used to submit it at the end of each year to the French government, and the French government used to give us back 30% of all our research and development expenditure, so that we can reinvest it again in the next year. Of course, this 30% did not come to my pocket, unfortunately, but it was very helpful for me that when I go, to the CEO, I tell him I would like to buy new equipment. I would like to, to participate, send some of my team to participate in a conference. He said that it's expensive. I tell him yes, but there will be 30% back from, from government. So this is helpful, to make the research and development happening in private sector and not only in academia. Next slide, please. So now if we talk about innovation itself, we can categorize it in different manners. And this is really depending on how we look at it. So we can talk about product innovation, process innovation, or business model innovation. Next slide, please. Another manner to categorize innovation, it's based on, next slide, please, the level of innovation. So we can talk in this case about incremental innovation. So for example, we have immediate release formulation of a drug, and we would like to make extended release or already dispersible. So we are improving an existing product. While another manner is to look at it is the radical innovation. So here we have talking about completely new class of drugs, like for example, CAR-T for oncology, or sometimes we are talking, we have the same, drug, but we have some radical transformation of this drug that make it really a radical innovation. So for example, if one day someone among you is able to make tablet insulin, this is really a radical innovation, because it will be much easier for patient to use and much easier to produce. Next slide, please. Finally, another manner to talk about innovation is the innovation approach. So we can talk about closed innovation, and in this case, everything is generated and developed within the same organization, while open innovation, the topic of our presentation today. So here there is collaboration between different parties to bring new innovative product to the market. Next slide, please. In this diagram, you can see the difference between these two approaches. In closed innovation, everything is done within the same organization. So we have generation of ideas, development of product, and the market. Everything is done within the same organization. While in open innovation, the generation of ideas is coming within the organization or outside of it. Development is done with collaboration with different parties, and there is the in-licensing and out-licensing process until arriving the best product to the market, and in our case, the best molecule, hopefully, to the market. Next slide, please. Now I would like to define with you what is open innovation. So open innovation is the ability of an organization to identify ideas for new processes, technology, products by looking outside its own environment. So open innovation is innovating with others for shared risk and reward. And I would like to emphasize on the idea of sharing both, because open innovation allow us to de-risk, reduce, mitigate the risk of the research and development, but at the same time, we need to share the reward. Next slide, please. Now, why we need open innovation in life sciences? I think the main reason is that there is a dramatic reduction of research and development efficiency in the last decades. So if you look in this diagram, you can see that in 1984, pharmaceutical companies in the United States used to spend about $9 billion each year for research and development activities. This amount has been increased currently to close to $90 billion. So it's tenfold increase. But unfortunately, this has not transferred in tenfold increase in the number of new molecules that we bring it to the market. So as you can see, FDA in the last 30 years always approved something between 20 and 40 molecule only. So based on that, the pharmaceutical companies has always looked to collaborate with others and to improve the efficiency of the research and development by having open innovation and working with others. So in the last years, about 70% of sales of new drugs that arrive to the market are not generated by the pharmaceutical company that bring them to the market. They are generated by other small company or academia. So this is, show the importance of open innovation in life sciences. So basically what happened is that academia, mostly they discover new target, develop new molecule, and then this new molecule, one professor of university with 2 PhD student or postdoc, they go, make spin-off, and then they use the incubator of the government. They use the technology transfer office of the university to raise a little bit of money to bring it to preclinical phase. And in preclinical phase, usually one pharmaceutical company become interested because there is clear proof of concept. They join the effort to bring it to clinical phase until phase 2 or even 3, and sometimes even a second pharmaceutical company come to help to bring it to the market. So the cost of bringing new molecule to the market is about $2.6 billion on average. But as you can see, different parties has participated on that. So the risk has been mitigated. And naturally, if it succeed and become, blockbuster product, every part from university until the big pharmaceutical company will share the benefit of that. So this is really what's happening, in the last years to bring new innovative drugs to the market. Next slide, please. Now talking about specifically of collaboration between academia and industry, there are a lot of advantages for both. So from university perspective, they can work in concrete, problems that can brought by industrial company and not only, theoretical problems for publications. They can leverage the project management knowledge from pharmaceutical industry. On the other hand, pharmaceutical industry can access a wide range of expertise from academia. So we have in the same area, medicinal chemistry, toxicology, pharmacology, which is very useful. Optimizing the cost of research and development, as we said, it's very costly to bring new molecule to the market. So especially in Europe and United States, there are a lot of funding coming from government to support the collaboration between academia and the private sector. So this is de-risking the cost of bringing new molecule to the market. Next slide, please. So what are really the hurdles that we need to overcome when we collaborate between academia and the pharmaceutical companies? The first one, I think, it's the bridging the culture gap. There is big difference how these two words function and look at innovation. So from industrial company, we are very much focused in teamwork, deliverables, metrics like timelines, budget, while academia is very much, about the reputation of the professor. So we say that this is the, laboratory of Professor XYZ. So it's a lot about person and reputation. Academia is very much motivated by publication. So the metrics of progress in the career in the academia is the publication. But in industry, we are making publication. Personally, I have published several papers, but we only make publications after we secure the IP. Okay, so the IB is our main target and publication come in the second place. Another culture gap that we have to look at it, if you can move the second one, please, is the disclosure. So working in open innovation, you need to be, very transparent with your partner. So you have to share knowledge, you have to share the information. But at the same time, as we said, we need to protect the IP. And so there's balance, we have to find it between transparency and protection of the IP and know-how of each part. Next slide, please. Now to arrive to the question, I receive it a lot from my friends and colleagues working in academia, how to land a deal with a pharmaceutical company. So first of all, you need to look at the business need of the pharmaceutical company. As you know, each company is working in specific disease area, oncology, neuroscience, infectious disease. And within this area, each company is working in specific indication. So you need to find out which company that might be interested for your specific innovative molecule. Another aspect you need to consider is that what is the type of your innovation? So some companies, especially the innovators company, they are not interested by incremental innovation. So if you have any improvement, any new formulation, for example, it's not what they are looking for. But on the other hand, generic companies, they might be very much interested by incremental innovation. So you have to address the right, business partner or industrial partner based on the level of your innovation. Finally, and this is what I really, find difficult to convey to my friends in academia, is that you have to have products and not idea. So if you have, very nice idea, very nice publication, it's not enough to go to industrial partner. Industrial partner would like to see a specific product with clear proof of concept. So they would like to see that it's not necessary to have clinical data. Okay, nobody expects that, but at least you have to have, very good in vitro testing, advantage and some proof of concept in preclinical models, so relevant preclinical models. So this will show that yes, as we said, the pharmaceutical company might spend up to $2.6 billion to bring it to the market. So before spending, all this money, they want to be sure that you have a good product. So this, you have to bring this proof of concept and not, ideas or publication. Next slide, please. So once you have, established which is your business partner and at which moment you are going to go to the business partner, then these are the steps that you need to go through usually to land a deal with a pharmaceutical company. The first one is non-confidential phase. And again, I find it in our region very strange that every time you talk with someone about, collaboration, first thing they tell you, let's sign confidentiality agreement. This is not how things going on. So we cannot, pharmaceutical companies, sign confidentiality agreement with, everybody they talk with. So you must be able to present non-confidentially your innovation. So for example, just one page that say that the IC of my molecule, IC50 of my molecule is this number. I tested it in preclinical model against, competitor and gave this result. No disclosure of chemical structure, no disclosure about how do you produce this molecule. Just, this simple information will be enough for the pharmaceutical company to say that, yes, this is something I am interested to discuss further, or no, this is out of my scope. Okay. So once you have shared this non-disclosure information and the pharmaceutical company say that, yes, I'm interested, then only we move to the confidential phase. Naturally, we start by signing confidentiality agreement. We might sign what we call it MTA, so material transfer agreement. So in order to take some, part of your molecule and test it internally, in order either to verify your data or to complete your data and see that if it's interesting to bring it to further phase. In parallel to this technical discussion, the business discussion initiated. So in order to define what is the model of the deal. So next paragraph, please. So for model of the deals, there are different models. So you have usually there are three components. First of all, upfront payment. So this upfront payment will allow to cover the expenses of the research so far. And then there are milestones. So milestones, it's, recognize the progress of the molecule from preclinical phase 1, 2, 3. And then hopefully if it's arrived to the market, then there's royalty on the sales. So these are the three elements usually in each deal. So now we arrive to the conclusion of this presentation. I would like to have some reflection with you about one important topic that can be, show the importance of open innovation. Currently, we have, something like 10,000 identified diseases in
Sorry to interrupt. No audio from the live venue. Yes, thank you, sir. You may continue.
Okay, thank you. So as you can see, we have only about 5% of diseases which has satisfactory treatment. So this is really a big health challenge for all of us. I think no single organization, private, public, no single country can address this global health issue. So we do need to work together in collaborative, open innovation manner to address this health need and this global challenge. And I think in the last couple of years, we have seen very nice, examples of open innovation with the COVID-19 situation. So we have seen that the genome of the virus has been decoded very rapidly, shared worldwide, and then immediately there was, collaboration between the university and industry, Oxford AstraZeneca, or between small pharma and big pharma, BioNTech, Pfizer, to bring new vaccine in less than 1 year. So this is really a good example. And I think we need to, yes, to follow in this, direction to have this progress and address this global health need by using an open innovation model. Thank you a lot for your attention, and I'll be glad to discuss with you during the day.
Thank you, sir, for your amazing keynote session. Yes, now I would request all our delegates at the live venue as well as the virtual, you may take a break. The duration of the break will be 15 minutes, after which we'll be continuing the keynote presentations. Thank you. Please be back within 15 minutes. Thank you.
hello. Just 1, 2, 3. Hello, hello, hello. Mic checking. Hello, hello. See? Hello, hello. Check 1, 2, 3. Hello, mic testing.
Hello, hello, hello. Hello, hello.
No, little more louder.
Hello, hello.
No, it has to be a little more louder. It's very low.
Hello, hello. Check 1, 2, 3. Hello, mic testing. Hello, hello, hello. Can you hear me? Hello,
We can hear you, but it's very low.
hello, hello. Sound come now. Hello.
Hello, hello, check. Hello, microphone check. Hello.
Yes, yes. That's a little more audible. That's fine.
Hello, hello, check. Hello, hello, microphone. Hello, check.
Yes.
Hello, 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 check. Hello.
Yes, it's audible.
Which one will be better? This one or last one?
The first one, not this one. The first one. The previous one.
Hello,
No, not this one.
hello, hello,
Yes, yes, it's audible now. It's clear.
hello, 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 hello. Okay, check. Now it's audible, right?
Yes.
Hello, hello.
The podium mic is not very loud and clear.
Hello, hello. Check one,
Yes,
two.
yes, yes.
Oh, hello. Hello, can you hear me? Hello.
Yeah, it's
Okay.
fine now.
Good. ABC. हाँ अच्छा अच्छा थोड़ा हाँ इतना हम बात कर रहे हैं तो साउंड में है ठीक है तो बोलो थोड़ा इतना डिस्टेंस करके बताओ ओके नाउ इट्स ओके कैन यू हियर मी
Little more front. When you come closer to the mic, I think it's a little more audible.
यस यस यस
That's what.
Welcome back all. I hope all are back after the short break. Yes, now I would like to invite our next keynote speaker, Dr. Razia Khanum, who will be joining us live from Dubai, professor and lead pharmacologist from Gulf Medical University, Ajman from UAE. She's a committed professor with over 20 years of experience at leading institutions in UAE and India, teaching students from various social and cultural backgrounds. Possessing excellent administrative, communication, and written skills, along with the constructive and effective teaching methods that promote a stimulating learning environment. Responsible for carrying out teaching using advanced teaching techniques, administrative and research duties, also involved in coordinating courses for PharmD, MBBS, and BBMS. I request Dr. Razia Khanum to deliver her keynote presentation. Welcome, ma'am.
Good afternoon, everyone. This is Dr. Razia Khanum. I am professor of pharmacology.
Sorry to interrupt. Ma'am, can you be a little more louder?
Yeah. So good afternoon, everyone. This is Dr. Razia Khanum. I am professor of pharmacology at Gulf Medical University, Ajman. And this is my first conference that I'm attending after Corona in person. And I'm sure the people who are here, it is also the same cause. So I'm thankful to the organizers that they have arranged this conference and invited me for the keynote speaking. And one of the theme of the conference was the new drug discovery. Being an academician, being a pharmacologist, and being interested in research, I'm always inclined towards this topic. And today I'm going to present some of the novel drug targets and their potential utility that can be used in hypertension. So now the question is that why hypertension I have chosen. Please, next slide, please. Next, one more. Yeah, so hypertension is one of the major contributing factors for the cardiovascular as well as for the renal diseases. Then it is associated with the risk of different comorbidities like heart failure, stroke, MI, etc. Various risk factors are there, including obesity, genetic factors, lifestyle disorders, and there is a very high prevalence. So if we talk about the prevalence, it is affecting globally around 1 billion people, and 9 million of the deaths are due to hypertension every year. Then there are various complicated regulatory networks that also affect the progression. And I'm going to take you towards the basics of a bit pharmacology because being in academics, this is my interest. So can I have the next slide, please? So I'm sure this type of slide, this type of picture or a similar picture we all have seen during our studies. So this is showing the classical targets of hypertension. There are around 10 groups of the drugs which are used for the management of hypertension that are targeting the renin-angiotensin system, autonomic nervous system, etc. Certain channels also like calcium channels. But still there is a need for the development of newer drugs because a considerable amount of a considerable population of people, around 10%, they suffer from resistant hypertension. Resistant hypertension is that despite using the drugs, either single or the combinations, then also the patient is not able to benefit. So next slide, please. So that these are certain newer drugs in the hypertension. The list doesn't stop here. There are few others also, but because of the time, I have selected these one which I found quite interesting. And here the drugs are already in phase 2 or phase 1 clinical trials. Some of them are still in preclinical stages also. So the first one would be anti-aldosterone agent that, and all of these I will be covering up briefly. Next, please. So these anti-aldosterone agents, we already have spironolactone and eplerenone, which are already in clinical use. but still there is a need for the development because of the adverse effect profile of spironolactone and less efficacy of the eplerenone. So the compound here that has been developed is finerenone, which is a non-steroidal antagonist for the mineralocorticoid receptor. It has a superior selectivity and affinity, and it has been proved to be better than spironolactone as well as eplerenone. So this is also similar to the spironolactone and eplerenone, but it is non-steroidal in nature. The other one is the aldosterone synthase inhibitors, which are going to prevent the synthesis of aldosterone, because there are certain limitations that these antagonists are having, like their effect on the cardiac and the vascular contractility and their sympathetic stimulation. So in this, the compound that is under development is LCI 699, which is the orally active aldosterone synthase inhibitor. So its studies has been done on the animal models. It has been shown to decrease the plasma and the urine aldosterone concentration, and the similar effects have also been seen on the humans also, and it is in the phase 2 clinical trial stage. Now the other drugs, because we keep on reading about the renin-angiotensin system drugs, so they are all peripheral. The central drugs are also they are also under development, and because it has been seen that the RAS, the activation of brain RAS, that also plays an important role in the occurrence and maintenance of the hypertension in the animal models. So here we have two of the enzymes, which are APA, that is peptidase A and N, which are responsible for the conversion of angiotensin 2 to 3 and 3 to 4. So the compounds here under development are EC33 and PC18. Next, please. So here in this picture, it can be easily seen where these drugs are going to be acting. So in the brain, they prevent the conversion of angiotensin 2 to 3 and 3 to 4. So angiotensin 2 and 3 are responsible for the pressure effects, and once they will be inhibited, their conversion will be inhibited. So that has been seen that the hypertension can also be prevented, or it can be managed. These compounds are still in phase 1 and the preclinical phase, and this RB150 is the prodrug of the EC33, which is the inhibitor of the APA. Next, please. The next one here is the vasoactive intestinal peptide receptor that has also been found as one of the potential target for the management of hypertension. This neuropeptide is able to exert positive inotropic, chronotropic, and the vasodilatory effect by its effect on the receptors VPA-C1 and VP2. These both are the GPCRs. So this VIP is associated with the various cardiovascular and the cardiopulmonary diseases. So hence, if this will make out and this will be available for the clinical use, it will be the target for not only systemic hypertension, but also for the pulmonary hypertension and the heart failure as well. So the compound here is Vesomira. I'm sorry, it is hidden with this panel. The compound here is Vesomira, which is in the phase 1 trial only. The trial names are also I have mentioned. They have been done, but the results have not been published yet. The next here is the sodium hydrogen exchanger inhibitors, specifically the isoform 3 inhibitors. So this sodium imbalance in the intake as well as in the excretion, that has been the key process in the pathogenesis of hypertension as well as its complications. So this NH3 is expressed in the intestines, and it plays a dominant role in the absorption of sodium, whereas in the PCT, that is the proximal convoluted tubule, it plays its role in the hypertension. So therefore, this NHE inhibitor, which is tinapanor, so that has been seen that in the experimental animals, that oral administration reduces the uptake of sodium, improves cardiac systolic dysfunction, and hence it can also be a potential target for the hypertension management. Now comes the vaccines. Recently only we have seen how much vaccines plays an important role. So here also the question comes that why not vaccines have been developed for such diseases that are lifelong and that can be that will be able to prevent the diseases. So we could have the vaccine even 50 years back for the prevention of hypertension. The renin vaccine was already developed, but it has induced autoimmune diseases of the to the kidneys, and therefore the further development was suspended. Angiotensin 1 vaccine and angiotensin 2 vaccines are already in different developmental stages, and their studies are already being carried out. Next slide can show you what is the present status of these ones. Yeah, so these are the various vaccines which are either in phase 2 clinical trial or the preclinical studies are going on. These are either the receptor inhibitors or they are the peptides, and they are the vaccines for, and they are inhibiting the either the receptors or these are the peptides like angiotensin 1, 2, etc. Next, please. Now, this is something that is very interesting. It is about the GI microbiota. So we all know how important is the GI microbiota for the health status, and this has also been seen to affect the hypertension pathogenesis. So in 2005, based on the metagenomics, a group of scientists, they divided the whole microbiota into 6 groups, and 2 of these groups which were the dominant, one were Bacteroidetes and the Firmicutes. So the ratio of these 2 can also reflect the degree of intestinal flora disorder. And if there is a dysbiosis, so that is closely related to hypertension. And the metabolites also play an important role in the blood pressure regulation. Next, please. Yeah, so it has been seen by different studies. They are present, they are available, well published, that how this microbiota is going to affect the hypertension, because the products of these microbiota, the intestinal microbiota, they ferment the dietary fibers into different products, which are the short chain fatty acids like acetate, propionate, butyrate, etc. Now, these short chain fatty acids, they will be either absorbed or excreted. So now these short chain fatty acids are acting on the different receptors like OLFR78 or GPR41, and modulation of these receptors by these fatty acids lead to hypertension. So this OLFR78, once it is stimulated, it causes an increase in hypertension, and GPR41 tends to reduce the hypertension. So what is the current status? Because we have lactobacilli, which is the predominant component of the human intestinal microbiota, and it also exerts anti-inflammatory activities also. Next slide, please. So this Lactobacillus helveticus has, which is the fermented milk only, that has been shown to contain certain bioactive peptides, which has the blood pressure lowering effect in the hypertensive subjects. Oral administration of recombinant lactobacillus, that also has the ACE inhibitory properties. And a simple study done by Ayogi et al., they have found that elderly people who took or who drink fermented milk products, this lactobacillus casei, at least 3 times a week, they had significantly lower risk of hypertension. So all these results indicate that lactobacilli can also play an important role in the development against, protective role against the development of hypertension, and probiotics will be having a major role to play there. But these are only the studies available. They are not into any of the clinical trial stages. Next, please. The last one that I'm going to discuss briefly here is the endothelin receptor antagonist. Endothelin itself is a potent vasoconstrictor. So therefore, its antagonist can also be one of the potential target for the management of hypertension. Endothelin acts on two type of receptors, ETA-AR receptor and ET-B receptor. Next, please. So we have here already different antagonists which are selective, non-selective for ETA receptor or ETB receptor. One dual antagonist is also there, which is aprocitentan. And at present, research on this compound in combination with the other drugs have been found to be useful in the treatment of resistant hypertension. The trial is already ongoing. And these results suggest that targeting endothelin receptor can also be one of the therapeutic target for the management of hypertension. So now what are the perspectives? The monotherapy in the treatment of hypertension may be ineffective as the clinical studies shows, and the new mechanism and the newer drug targets discovery that itself is challengeable. So many of the companies, they have started focusing on developing the fixed dose combinations, and various fixed dose combinations are being tried, like ACE inhibitor with diuretics, calcium channel blockers with the other first line drugs like angiotensin receptor blockers, etc. So these dual or triple drug targets, these are also the agents that have been tried for the management of hypertension, including the resistant one. Next, please. So in conclusion, as we all know that it's a common risk factor for the cardiovascular disorder. So various studies are required for the management. These findings include insights into the mechanism. They can help in facilitating the development of the drugs. Various steps are required for the development of drugs. First is that the targets must be screened. Second is that the appropriate and the personalized daily doses of the available drug must be clarified. And finally, the adverse effects or the side effects that also that can lead to the organ damage, they must be also finally resolved. So these are all the references, and thank you so much.
Thank you, ma'am. Thank you for your amazing keynote presentation. Any questions? The participants present at the live venue may ask the questions, or if anybody from the virtual mode, if you have any questions, you may put in the chat box. Okay, I guess there are no questions. Thank you so much, ma'am. Yes, yes, please, any questions?
Good afternoon, ma'am. Dr. Ranjan, what is your name actually? Okay, I think she's doing. She has given a new input on the cardiovascular diseases, and I have one question for her. Actually, how do these new targets override the existing therapy for cardiovascular diseases?
Dr. Razia, is the question audible to you?
Can you please repeat again one word I didn't?
Yeah, one word. Actually, the new targets, according to you, the new targets, how do the existing therapeutics available for cardiovascular diseases override the new targets?
Yeah, so we have around 10 group of drugs that are already available for the treatment, but still we see the comorbidities and the resistant cases, and that is why there is a need for the development of the newer drugs. These newer targets or the newer drugs may be helpful in managing the comorbidities, the cardiac remodeling, the vascular remodeling, as well as the resistant type of cases. They can be managed by them. And these drugs are still in pipeline. They are still in the different stages. But we expect that some of them were in clinical trials for the treatment resistant hypertension only. So that is why it is expected that if they will at all be there in the market, so they can manage those cases as well.
Well, regarding you were mentioning about the vaccines, right? So for vaccines for cardiovascular diseases under development in preclinical stage, so how could because mostly vaccines are actually for infectious disorders, and as I know, my knowledge says most of the vaccines are for infectious disorders, and especially for cardiovascular diseases vaccine. Maybe any morbidities develop later, or it would be successful in your view? What do you think?
Initially, the one that was able to be able to reduce the blood pressure in the animal models, that was found ineffective, and the other one was found to produce autoimmune disorder in the humans. So that is why those studies have been suspended. Recently, what is going on, I have shown there that these drugs are not only targeting renin, but angiotensin 1, angiotensin 2, as well as the receptor blocker they are. So one of them on the receptor blocker, that only is in the phase 2 trial, and the rest others are only in the preclinical stages. They are either the receptor blockers or the peptides by themselves.
Peptides, yeah, correct, yeah. Thank you, ma'am. Nice presentation.
Thank you, Dr. Razia, and thank you, Mr. Lokesh, for your question. So I hope there are no further questions for Dr. Razia. Thank you so much, ma'am, for your amazing presentation. So next, we will quickly move on to a group photo presentation. I would request the live participants who have joined our delegates, keynote speakers, to kindly join for a group photo. And our participants who are live on the Zoom platform, you may enable your camera and we may take a group photograph in the Zoom platform. Yes, thank you so much. Yes, thank you all who have joined us on the virtual platform for the group photographs. Thank you so much. So next we'll be inviting one of our presenter who is ready to give a presentation at the live venue. I would like to invite Mr. Sajid Majid Hamid, who will be discussing about pharmacist role in antimicrobial stewardship program application in pediatric hospital. He's from Saladin Health Directorate from Iraq. Welcome, Mr. Sajid Majid. Yes, Mr. Sajid, are you ready for your presentation? Okay, next we'll quickly move on to our next presenter who's available here, Dr. Inam Sameh Harif, who will be discussing about the synergistic effect of arsinoid X receptor and fasting mimicking diet against metastatic castration-resistant prostate cancer. Dr. Inam is from College of Pharmacy from University of Mastan Risa from Iraq. Dr. Inam, welcome. Requesting technical team from live location to connect Dr. Inam's presentation next. Sorry, team. We'll be connecting with our keynote speaker who has joined us virtually. Meanwhile, we'll get back to the live location. Next, I would like to invite our keynote speaker, Professor Marcel Muzaffri, the president and founder of Australian Nanoscience and Nanotechnology Initiative. Professor Marcel Muzaffri graduated from the School of Pharmacy and Chemistry from Liverpool John Moores University in 2005, and he has authored more than 100 publications, including 3 patents and 3 books, which are among the first books in the field of pharmaceutical nanotechnology and nanobiotechnology. His book, Nanoliposomes From Fundamentals to Recent Developments, is the very first book on nanoliposomes ever written. Professor Muzaffri possesses 20 years research and teaching experience, both in the academics as well as the industry in different countries, including England, Turkey, New Zealand, and Australia. I request Professor Marcel Muzaffri to deliver his keynote presentation. Professor Muzaffri, can you hear us? The video from the live location is off. Can we begin? Mr. Sajid, the audio is not audible to us. Sorry to interrupt, but the audio is not audible from the live location. Team, can you please check the audio and video from the live venue?
Yes. Yes.
I will confirm you. I will buy for ticket
Thank you, Mr. Sajid, for your presentation. Yes, and we are being joined here by our keynote speaker, Professor Marcel Muzaffari. Good afternoon, sir. Thank you for joining.
Good afternoon.
How are you doing, sir?
Good, good. How are you?
Good, sir. Thank you so much. Sorry about the technical glitches, but you stayed on with us for so long. Thank you so much, sir. And you may begin your presentation, sir.
Yes, I have the presentation on my second screen.
Okay.
Now, if you guide me how to share the screen to
Yes,
the
sure.
audience.
In your Zoom account, do you see at the bottom share screen a green color button?
Yes.
You may click on it.
It needs your permission.
Yeah, just a moment. Requesting the host to give the keynote presenter the co-host access.
Yes, it says host disabled participant.
Team, can you hear me from the live location? Can you please give access to Professor Muzaffari? Yes. Can you please check now?
Okay. Okay. No.
Are you able to share screen?
It says open system preference.
Yes, just click on the tab where you have opened your PPT. Just click on it.
Yes. It took me to security and privacy.
So you just have to click on the share button.
I have I'm repeating the process again. I clicked on share screen.
Yes.
And
then do you see the files that you have opened?
I can see 8 options: desktop 1, 2, whiteboard, iPhone pad via airmail, another iPhone, universal access, preview, and Google Chrome. I don't want Google. I want a PDF file to be open. Now, how to do that? So I am clicking on desktop 1. It takes me to open system preference.
So can you just quickly WhatsApp or email us your presentation so that we can share the screen for you?
Right. I can email it to you. Please give me an email address.
Yeah, sure. So to the Pooja.
Okay. At techno.
Yes, yes. Technorate.org.
Right. I give the title presentation And sorry for the sake of time limit with no salutation. So I just send you a blank email with attached file.
Okay, okay.
And it is a PDF file. I hope the size is not too cumbersome.
Okay, okay, no problem. We'll get it done. So meanwhile, we are opening Professor Muzaffari's presentation. We can take up some general questions that the attendees would like to ask Professor regarding the recent studies that maybe he's doing now. So can you please discuss about something that recent activities that you have been involved in?
Right. So recently we launched our number 14 patent application.
Mm-hmm.
So these patents are generally in the broad area of biotechnology, and to be more precise, nanobiotechnology. I have not been limited to teaching and academia. I was privileged to have opportunity to work with industry since roughly 2010. So far, I have launched more than 10 nanotechnology products to the market using niche and advanced technology. And it may be interesting for the audience that I fully respect environment, and the methods I use for these nano formulations is called green technology. I will briefly explain during the presentation that these nano formulations, basically, we make the conventional therapeutic agents, drugs, vaccines, diagnostic agents, gene therapy protocols, nutraceuticals, vitamins, and immune boosts, which are on high demand currently because of the epidemic. And somehow we engineer them to be smart technologies in the sense that we increase their bioavailability. So the problem of solubility will be solved by choosing right formulation. Bioavailability will be enhanced by minimizing elimination and guiding the therapeutic agent where its action is needed in the body. We are not limited with any particular type of drug administration route. It can be injectable, IV, IM, it can be ocular, it can be transdermal. So we have been working with different types of formulations, including aerosol and pulmonary and nasal drug delivery formulations. Excuse me, just refresh my throat.
So where do you see the future of nanobiotechnology in pharmaceuticals?
One of the slides I want to share, the monetary aspect of the technology is mind-blowing. The numbers are hundreds of $billions annually. This is how much global market are paying. I got an email. Okay, so you received the presentation. That's good. The market is thirsty for health benefit products. After the pandemic of 2019, consumers now are more aware of the importance of high technology formulations.
Okay, I think, yes. I think your presentation is ready here, sir.
Right now I can very nicely link my current conversation to this one.
Yes.
Consumers are thirsty for new technologies. One of the technologies which can make current therapeutic agents smart is invented by us, and we gave the name tocosomes. So the topic of my presentation today is tocosomal drug delivery systems, and they are safe and efficient techniques based on tocopherol phosphate, alpha tocopherol, vitamin E. Now, how can I shift to second slide, please?
Yes, sir, we'll move it here.
Thank you very much.
Yeah.
And this topic is presented within the scope of 9th World Conference on Pharmaceutical Science and Drug Manufacturing. If I am not mistaken, if my memory allow me, this is maybe interesting for your self-organizing committee and audience. This is the very first time the subject of tocosome being presented at a conference. So for the first time in the history of mankind, tocosomes are being explained not on the paper, but as a lecture. So they are a technology used for drug delivery. What is drug delivery? If we search drug delivery online, we can see a brief description. It refers to approaches, formulations, techniques, preparation methods, storage systems involved in transporting a pharmaceutical compound. I add to pharmaceutical also food and nutraceutical and cosmetics. So application is dramatically and fastly broadening. We are not limited only to pharmaceutical industry. Currently in Thailand, we are also aiming to manufacture nutraceutical products, not only for human, but also for pet and livestock, to their target site in vitro and in vivo. Next slide, please. Now, this is the number I tried to remember and mention. I was right. I mentioned hundreds of billions of dollars. Yes. So this is a slide I put in one of the groups I am managing on LinkedIn called nanoliposomes, that it is envisaged that by 2025, 3 years almost ahead of us, the demand for drug delivery systems will reach almost 100 billion US dollars. So this is one aspect of the importance of these technologies. Available technologies, next slide, please, comprise to something like 100 different encapsulation systems. If we narrow down this number of carrier technologies, drug delivery technologies, to lipidic, as I call them, one category would be lipidic carrier systems, which include archaeosomes, cochleates, etosomes, liposomes, lipospheres, nanoemulsions, nanoliposomes, phytosomes, solid lipid nanoparticles, tocosomes, transferosomes, vesicular phospholipid gels, and you see tocosomes are among these technologies. Next slide, please. And this list is available in our paper, which incidentally has been the most cited and the most viewed paper ever published in the journal Pharmaceutics all time. You can see I circled in purple and designated it with a blue arrow, that not only last 6 months or last 12 months or last 3 years, in the whole history of these journals, our paper ranked #1 with respect of the number of times being cited. If you go to Google Scholar, the citation is more than 1,000, but they limit to PubMed. And the table I showed in the previous slide is from this publication. And almost every day, 2 or 3 new papers are citing this paper. Next slide, please. Right. So tocosomes were first introduced in 2017 in International Journal of Pharmaceutics as a rapid communication. You can see the phrase rapid communication under the logo of Elsevier. Because of importance of the topic, journal decided to publish it, avoiding the normal queue. And it is a 2-page paper for the first time ever mentioning in written tocosome. And I reiterate, Today, this topic, tocosome, invented by us, is being spoken about, being lectured internationally for the first time. Next slide, please. So what are tocosomes? We can compare it with an older technology which is known as nanoliposomes. Audience may know that the first book ever written on the topic of nanoliposomes was authored by myself. So the book Nanoliposome, published in 2000 and I think 2005, if I remember correctly, was the first book on the subject of nanoliposomes. Tocosomes were launched in 2017, much more new technology. They have similarities, these two techniques of drug delivery, and they have differences, which I will be explaining shortly. Next slide, please. Tocosome is a colloidal and vesicular bioactive delivery system. When we say bioactive, we are using the word very broadly. It can be vitamin, mineral, vaccine, diagnostic agent, genetic material, cosmetic compound, cosmeceutical, food, and nutraceutical. The main constituents of tocosomes are phosphate group bearing alpha tocopherols. However, like nanoliposomes, they can also include sterols as their ingredients, not only alpha tocopherol derivatives, but also sterols like cholesterol, proteins, and polymers in their structure. The phosphorylated form of alpha tocopherol is known as alpha tocopherol phosphate. I designated in yellow font on the screen. This compound is naturally present in human and some animal tissues. So it is not an alien compound. It's not toxic. It's completely natural, and we have it in our body. So we use ingredients from our own body to make capsules, so to rectify the concern of toxicity and using exogenous material. It has recently been reported that the TP molecule, that is tocopherol phosphate, is naturally present in certain fruits, green vegetables, cereals, dairy products, as well as in different nuts and seeds. TP is composed of a phosphate group attached to one hydrophobic chain known as phetyl tail, made of 3 isopropene units. On the other hand, D-alpha tocopherol phosphate, T2P, is a closely related molecule to tocopherol phosphate is composed of 2 phetyl chains. So TP has only 1 chain, T2P has 2 chains. Like those who have been working with liposomes, they know that the ingredients of liposomes and nanoliposomes are mainly phospholipids. And phospholipids, they have 2 fatty acid chains. T2P also have 2 chains, but they are not fatty acid. The chains are called phetyl tails, whereas TP has only 1 chain. Right. Unlike phosphatidylcholine and some other phospholipids, the hydrophobic phetyl chain of T2P cannot align in parallel. This will be shown in the next slide. Due to the presence of bulky side group. You see the angle I designated at with the Greek letter, what was it? Is it delta? I forgot. Anyway, I'm losing my Greek knowledge. There is an angle between the 2 tails of T2P at the bottom of the figure. So on the top, we can see chemical composition of tocopherol phosphate and 3-dimensional presentation of TP. The tail is shown in green color, phosphate group is in dark color with the letter P on it. And down, we can see that 2 molecules of TP are joined by 1 single phosphate group to constitute T2P molecule. Next slide, please. So we now compare TP on the left side of the slide, alpha tocopherol phosphate, with a typical phospholipid. As I mentioned previously, phospholipid molecules generally have 2 fatty acid tails. So the geometry of these molecules can be conical, can be cylindrical, can be inverted, conical, so on and so forth. And these tails are connected to a phosphate group on their head by the mediation of a glycerol molecule. When we compare it to the picture on the left-hand side of the slide, we can see that TP has only one chain, and that chain has 3 side chains, CH3s, on the left-hand side of the picture. And the chemical name is phetyl tail, whereas the chains in phospholipid are known as fatty acids. Next slide, please. Now, clinical investigations have shown that TP and T2P, the molecules we use to make tocosomes, they have many health benefits, including anti-inflammatory, atherosclerotic preventing, cardioprotective properties. Furthermore, the inhibitory effect of TP molecule towards tumor invasion has also been reported. Studies also show that alpha tocopherol phosphate protected primary cortical neuronal cells from glutamate-induced cytotoxicity in vitro and reduced the levels of lipid peroxidation. So we are making capsules, even with nothing inside them, nothing encapsulated inside them. The empty tocosome has a lot of health benefits, as listed in this paragraph from the reference cited below the slide. Next slide, please. Right. Tocosomes, nanoliposomes, liposomes, and a list of more than 30 different drug delivery systems can be made using methods which can be grouped broadly as safe methods, which do not use toxic solvents or detergents or extreme pressure or pH, and conventional methods. Methods such as thin film hydration method on top of the table on the left-hand side, you can see the name of the method, necessitates application of potentially toxic solvents. In the final product, even if we subject them to 24-hour vacuum, we have reported in the German pharmaceutical journal Pharmacy that is still the liposomes made by using thin film hydration method are significantly toxic to the liposomes made by heating method or Mozaffari method. Mozaffari method uses only one pot. It is a one-pot technology, one vessel, especially in industry. We don't like to move the product from vessel number 1 to vessel number 2 to vessel number 3. We prefer to put the ingredients in the first vessel and get the product from the same vessel. This is achievable by Mozaffari method. And there are about, I think, roughly 40 publications from different countries in the world explaining my method for different applications. Next slide, please. Right. So to sum up what I have been telling so far, among the vast number of bioactive carrier systems, drug delivery systems, nanoliposomes and tocosomes are the most promising. Why? Simply because they are safe. Their ingredients not only do not harm the body, but they are beneficial to consumer, being human, pet, or livestock. They are economic, so the cost effectiveness is also there. They have acceptable shelf life, generally well more than 2-year shelf life of the products. And they can be used for different applications, as I have mentioned before, for the prevention and diagnosis of disease, for nutraceuticals, immune boosting, vitamin, mineral encapsulation, and also now we have applications of these products in skincare and cosmetic products. They are not limited to water-soluble agents. They can also be used to encapsulate amphiphilic compounds and lipid-soluble compounds. Now, to encourage the audience and young researchers, the next slide is our recent patent, please. Can we, right. This is 2021 Australian patent. We use the mentioned technologies for the fortification of solid and semi-solid food products. In future, the future inventions will be limited to the field of pharmaceutical, but this year we focused on food. And the next patent, same technologies are used. Next slide, please. A couple of months after that patent, we launched another patent, which is now granted. Next slide, please, with the title of Method and Composition as Green Tech Preservative. So one of the other applications I forgot to mention is mentioned here, preservatives for pharmaceutical product as well as food product. No matter how good is your pharmaceutical formulation or nutraceutical or cosmetic product, you need to make sure it can be stored well above 2 years after manufacture, after being packaged. In most of occasions, we need FDA approved and safe preservatives. So this patent, which is now available online, explains how we use technologies such as nanoliposomes and tocosomes for manufacturing of acceptable green tech and safe preservative for food products, which can also very well be applied in pharmaceutical industry as well. And next slide, I stop here and I will be happy to take questions.
Thank you, Professor Muzaffari, for your amazing presentation. That was a lot of information. So thank you so much. Any questions for our keynote speaker?
You can type. Or
yes, you may type or ask the question. And if anybody have any questions from the live venue, please let us know.
Good evening to all. Actually, Malaysia, we are good evening now.
Good evening, sir.
Actually, I need to rejoin from my office. Actually, no, I just stay away for a while to drive back to home, and I'm at home. Prof, I just now saw your molecule, which you prepare. I would like to ask one question, that you have got both the hydrophilic core and also the lipophilic core, like for example, tocopherol. So how do you manage the co-solvers actually when you prepare the nanoparticles, nano size?
How to manage solubility?
How do you manage solubility? Solubility, I can say,
right.
Simply.
Right. So a brief overview of the methodology. We either are aiming to encapsulate water-soluble reagent or amphiphilic or lipophilic. For amphiphilic and lipophilic compounds, the procedure is pretty much the same. For hydrophilic compounds, they will be put in the main medium, being a buffer, a saline solution, isotonic solution, or just deionized water. So we take the vessel, put the aqueous component according to the formulation we intend to manufacture, then put the water-soluble molecule, increase the temperature based on the proper physicochemical properties of the bioactive agent. For Muzaffari method, temperature does not exceed 70 degrees Celsius. And only genetic material so far among the all molecules that we have been dealing with and launching to the market are sensitive to 70 degrees. And we have modified the technology and published it. We can encapsulate nucleic acid molecules, including mRNA, at 40 or below degree temperature. Back to your question, for lipid-soluble molecules, so we have two phases. We have aqueous phase in the vessel, and we have in a bucket outside the vessel. I'm now here picturing the situation in industry. In that bucket, we have the ingredients, TP, T2P, which are amphiphilic. They have a hydrophilic head group and a lipophilic tail, one or more lipophilic tails.
Yeah, yes, yeah.
So the lipid-soluble molecule will be mixed with those amphiphilic molecules. And we use a co-solvent, which a general candidate as a co-solvent, and this person would be glycerol or any other polyol, right? We will give them a very mild mix just to homogenize the mixture of the ingredients, including the lipophilic molecule. And then once the agitation, speed of the mixing and heating reached what we are looking for, between 40 to 70 degrees, we introduce the lipidic phase at the same temperature to the bulk component, bulk vessel. And the mixing, without the need to use any organic solvent, will help the amphiphilic molecule to arrange themselves into the shape of two layers of TP2P or phospholipid molecules. And that is our lipidic phase. It can be unilamellar, or it can be like onion, multi-layers of amphiphilic molecules, and the lipid-soluble molecule will be accommodated, placed in that lipidic phase.
Okay. I mean to say it is like you have a biphasic system in the single part, because you mentioned that single vessel.
It is a single part technique.
Single part, yeah, single part.
Yes.
We got single part actually, no, single part synthesis, yeah. Yeah,
because
that's
even with
interesting.
single part, you need another vessel to put your ingredient inside.
Yeah, understood. Yeah, it's something like you're making, yeah, understood. It's interesting. Sounds interesting, actually. I think we can apply for both lipophilic molecule and amphiphilic molecule together. Hydrophilic molecule together.
Easily.
Or maybe there is one more method. You can do conjugation as well. You can do conjugation as synthesis. Then you can separately prepare in nano carriers. Thank you, Prof. Thank you.
If conjugation does not jeopardize the activity, the biological effect or pharmaceutical effect of the molecule, yes, we can use that approach.
Yeah, because mostly I think you have ester bonds, and mostly I think it looks like ester bonds, right? Because hydroxyl and carboxylic acid ester bonds, or maybe either bonds, and which can break down by the enzymes, esterases within the biological system. So
as we make sure the desired effect is not damaged.
Yeah. That's right. Thank you, bro. Thanks. Thank you, bro.
Thank you for your question.
Thank you, Dr. Lokesh and Dr. Muzaffari, for your amazing inputs on this topic. So next we will be connecting live from the Dubai location, where we have our next presenter. I would like to welcome Dr. Inam Sameh Arif from College of Pharmacy from Mustansriya University, Iraq, who will be discussing about the synergistic effect of farnesenoid X receptor and fasting mimicking diet against metastasis, castration-resistant prostate cancer. Requesting technical team to connect the audio and video from the live location.
سلام آقا میثم.
سلام آقای دکتر، وقتتون
خوبی
بخیر.
عزیز؟
خسته نباشید.
قربان شما. من
عذرخواهم بعد موقع مزاحمت شما آقای دکتر، واقعا عذرخواهم، در جریان نبودم.
تعارفو بذاری کنار، خواهش میکنم.
ماسک کن.
من الان 2 تا مانیتوره، یکیش این کنفرانس دبی ادامه داره، ولی حس میکنم شما خیلی عجله دارید و اون زمان من در خدمتم. بگو پسرم، بگو.
آقای دکتر اگه شما کار دارید بعدا تماس میگیرم خدمت.
نه بعدنی وجود نداره، با خدا هر روز درگیرم. خداش منم اینه پدر.
عذرخواهم شرمنده آقای دکتر.
منم اینه پدر. ببین نگوین.
آقای دکتر منو
Next, please. Of the genes responsible for the proliferation and migration. Prostate cancers associated with decreased level of P10. By the use of INT-747 agonist, the P10 level increased significantly, and this increase was enhanced by the use of the media which is deprived of glucose. This is a protein which is responsible for regulating the proliferation of the cells. The other factor, cyclin D, regulates the the migration of the cell. The cyclin D usually pushes the cell from passing from one phase to another, especially the S phase. Next, please. Next slide. So, from these results, we can conclude that the INT-747 agonist drug, it decreased the proliferation, it decreased the migration of the cell, it enhanced and induced apoptosis in the cancerous cells of the prostate. On the other hand, we found that the P10 level of this protein have been increased by the use of the different approaches of the treatment, INT-747 and the glucose-deprived median. At the same time, this was associated with decrease in this level of cyclin D1, which is responsible for the migration of the cell. So we do, from these results, we do agree that there is a possible crosstalk between the activity of the FXR receptor and the metabolic activity, which is controlled by the type of media and type of diet that might the patient receive. Thank you very much. Any questions?
Thank you so much, Ms. Enam, for your presentation. Any questions? Okay. Okay, thank you so much. Next, we'll be going on for the lunch break, requesting all the members at the venue at Holiday Inn to join for the lunch, and the presenters who are waiting at the Zoom platform, virtual platform, you may also go for a lunch and be back by another hour. So the lunch break duration is 1 hour, requesting everyone to be back after 1 hour.
another
قیافشون رو میخوام ببینم. من ما رو نمیبینم. تو اینو میزنی خاموش میشه. الان میخوام صداشو بشنوم.
Hello.
Hello, good afternoon. This is Dr. Anand Shah.
Good afternoon, sir.
Good afternoon.
Thank you for joining.
Yeah.
Yes.
Pleasure.
So welcome back, everyone, after your delicious lunch break. So now we'll be moving on to our keynote presentation, and we have been joined here by Dr. Ranendra N. Saha, the director of Shri B.K. Birla and Srimati Sarla Birla Chairperson, BITS Pilani, UAE. Dr. Ranendra Saha has been a senior professor of pharmacy and director of BITS Pilani at Dubai campus. And in 2011, he has been awarded as the Shri B.K. Birla and Srimati Sarla Birla Chair Professorship at BITS Pilani for contributing in teaching and research. He has vast experience in the field of pharmacy, especially in pharmaceutics, novel drug delivery systems, and pharmacokinetics. He also received Pharmacy Professional of the Year 2013 award given by Indian Association of Pharmaceutical Scientists and Technologists. He is also a recipient of the Best Pharmacy Teacher Award in the year 2005. He has more than 33 years of teaching, research, and administrative experience. He has published more than 90 papers in international national journal and presented more than 100 papers in conferences, seminars, symposium. He has also delivered several invited lectures at seminars and conferences and universities in India and abroad. Thank you, sir, for joining. I request Dr. Ranendra N. Saha to deliver his keynote presentation. Welcome, sir.
Thank you, Jajila, Ms. Jajila, and thanks to the organizer for inviting me to deliver this keynote lecture. It is my pleasure. I will speak on, I'm sharing my screen. Can you see my presentation copy?
Yes, sir. Can you put it on slide mode? Yes, it's visible.
Yeah, sure, I'm doing that.
Yeah.
Yeah, is it okay?
Thank you, sir. Yes, yes.
Okay, fine. I will speak on drug delivery, but what are the challenges of conventional drug delivery and ways to selective delivery for better therapeutic efficacy? That how it can be done, and really what we did in our research lab, in our group. I'll try to present some data within that 25 minutes, and I assure you that I'll not take more than 25 minutes. I'll finish with that one. You know, new drugs coming to the market is very, very limited. Many reasons are there: the cost, time, and then regulatory requirements, challenges, side effects, etc. So question, a very, very important question and challenge to the formulation pharmacist, that how will we modify the formulation so that we avoid many of the existing problems like side effect, toxic effects, excessive cost, etc., for which the novel delivery system can be very, very useful. So it is very, very important to design better drug delivery systems for better therapeutic efficacy. And it is possible because new insight of diseases and advancement in pharmacokinetics studies made the drug delivery totally different and more logical and scientific. It is possible to achieve selective delivery for better efficacy. And nanotechnology can be very, very helpful, which I'll try to prove. Some of our published data done at our group in our laboratory at BITS Pilani. You see, the problem of selective delivery is that many drugs are having non-selecting delivery. It goes to all part of the body. A simple way I try to explain that, if assume I want to target a drug to brain, maybe anti-cancer drug or some other drug, but only 20% of the drug goes to the brain, and I need 20 milligram to reach brain to get my required action for a required period of time. Now, if it is only 20% goes to the brain, for 20 milligram drug, I have to administer 100 milligram, and then remaining 80 milligram is going to the other part of the body, which can cause side effects on those parts, a cardiotoxicity or other toxicity is possible, because 80 milligram drug is going So 4 times the drug going to the brain is going to the other part, and patient has to bear the cost of that total 100 milligram, though patient needs only 20 milligram. Now think of it, that if you can make a delivery by some process where 50% of that drug can go to the brain. Now you can see, to get same 20 milligram, you only need 40 milligram dose to be administered, because 50% of that dose administered can go to the brain. What happens then in that case? 20 milligram goes to the brain, and out of 40, only 20 milligram may go to other parts of the body, which is lowering the dose from 100 to 40, and this 20 milligram may not be able to produce any side effect or toxicity. This is, I am saying, is a selective delivery, so that the drug can go maximum to the site of action. I think this is a very, very important aspect you can see. If we can make, particularly think of anti-cancer drug, which you know causes very severe side effect toxicity to the patient. Why? Because the drug is non-selective in both action as well as non-selective in distribution. So if we can achieve that, it can be very, very useful for the formulation point of view, patient point of view, better efficacy, avoidance of side effect, toxic effects, as well as better patient compliance. So better drug or not delivered, novel drug delivery can help in targeted delivery at the site of specific action. So it will not be producing any action not available to other places. Or it can be selective distribution or modified distribution, more towards the site of action, as well as controlled release, so that you do not require to administer multiple times. This is a unique benefit we can get it if we can design on a novel drug delivery system. I'm simply trying to explain it in this diagram. You can see when we administer a formulation, after the drug administered at site of administration, whatever form may be, solution or something else, drug is absorbed from the site of administration. After dissolution, it is absorbed, coming to the systemic circulation, and goes to the site of action and other sites. This other site is the problem. If very less goes to the site of action, maximum goes to the other site, which can cause side effect, toxic effects, what not, and cost is also increasing. But you think of it, if you can make something that the drug goes to the site of action, but maximum, but not to the other site, you can achieve many, many benefits, and it will be better for the patient also. You look here, same thing I am trying to explain in another way. The central compartment is coming and then going to the target organ, where the drug is binding to the receptor and produces the action. But other parts of the body, even something going, if that is not available there, it cannot produce any side effect, toxic effects. So the challenge is to how to deliver the drug to the site of action selectively, but not to the other places. Maximum, so that those can be reduced. Obviously, whenever I do, it is an important, how do I judge it? How do I evaluate it? Say, for example, I am claiming I have made a novel delivery system, which will be selective. You also claiming the same thing. How do you compare between me, my product, and your product? It's a simple way I'm trying to explain. Many other methods are there. It is basically to give you the idea that why and how we can evaluate novel delivery system. You assume S is the system, and drug is required to be at the site of R where the receptor is there, and T is the other place where the toxic effect happens. When conventional dosage form, you can understand from the system, a little amount of going to the R and maximum amount going to the T or remaining in the S. Now in that case, I can get the area under the curve at the R and area under the curve of the T. If I take the ratio for conventional dosage form, AUCR is going to be very less, AUCT is going to be higher. So probably it is less than 1, the fraction. The denominator which you are saying it, AUCR by AUCT for conventional, is a less than 1. Because AUCR is lowest. But if you can make a novel delivery system and administer that and measure the AUCR and AUCT, the ratio of the two is supposed to be increased. So the numerator ratio and the denominator ratio, the ratio is more higher, the product is more selective distribution. Now by this process, my product and your product can be compared, and if the ratio of the final ratio is maximum for your product, that means your product is better selective than my product. This way, the important part is that how we can decide a dosage form, whether it is a better selective distribution or not. We have been able to achieve that which I'm trying to explain to you with some result. So nanotechnology, what we have worked is using the nanoparticulate system, which is very, very useful for potential in therapeutics. It has many advantages. It provides many benefits. It can stabilize the drug in vivo. It can facilitate better absorption, better availability, modify distribution, and enhanced PK. When I say enhanced PK, means pharmacokinetic parameters like elimination rate, half-life, etc., can be changed by making the nanoparticles, which I'll show. It can also help in controlled release of the drug for increased therapeutic effectiveness, so that delivery can be reduced or frequency can be reduced. Drug targeting is possible, selective distribution is possible, modulation of the special drug distribution or selective distribution, more efficient utilization of the drug by this process, so the cost can be reduced. And obviously, improve patient compliance, which is the most important part. Think of it that today for anti-cancer drug to a cancer patient, if you can make it selective to the only cancer site, how much benefit it can be. Same thing for the drugs which are going to brain, other drugs, CNS active drug, etc., which sometimes have a cardiotoxic and other side effects. So these are the various benefits is possible. So we also worked on nanoparticulate system, liposomal system, which at least for 2 drugs, I'll share the data that what we have, I am saying that it is possible to achieve it. And it is possible to get better therapeutic efficacy, which we have done on animal model and started. We have worked on 2 drugs. The first drug actually basically for another drug, we worked, but then initial study we did it dopamine. For that dopamine central nervous system, its permeability to blood-brain barrier is a very, very poor. And that's why it is normally the prodrugs of dopamine which are given, administered to the system. What we tried to do on the first drug dopamine case, we tried to deliver it nasal route to see that selective and efficient delivery is possible or not. And on the basis of that study, we have worked on another drug for the company which sponsored the project and delivered them a liposomal preparation, which has been patented and taken to the market also. And I'm fortunate to say that I have multiple drugs which have been marketed in Indian market. Controlled release product, Movon CR, Gidorol CR is developed at Bitsmillani lab and marketed in India also. Okay, what I'll present is I have 2 drugs I'm presenting here. One is the dopamine, other is the olanzapine. I hope you are able to hear me properly. Is that okay? Hello? Ms. Jejila, I hope I am audible.
Yes, sir. Yes, sir.
Okay.
You're audible, sir. You
Fine.
may continue.
Because occasionally, virtual meeting, that is an important. Where suddenly sound is problem or not. Occasionally checking.
You're audible and clear.
Okay. Thank you. We made liposomal preparation of dopamine and different liposome, 3 different types of liposome, mainly prepared, but 3 I am presenting it here, and delivered it nasal route to the rat. And what is the result we found? You can see here. The bottom line, if you see that near the zero origin is the drug which is given simple solution of dopamine through nasal route, and you can find that the drug present in brain is a very, very less. It is a very, very nominal, you can see. This is what we have done it, by the way, in that experiment. We first made the animals dopamine deprived, the dopamine total. We administered a substance to stop the dopamine secretion in the brain. So we created the animal which is dopamine deprived animal, and then we administered so that our analyses are interfered by the natural dopamine present in the brain. So when we administered, then we sacrificed the animal and estimated the drug concentration or amount in brain at different time and plotted against graph in here. You can see concentration versus time. So you can see area under the curve or the concentration reached a very, very low when it is given in solution form. But when it is given in liposomal formulation, you can see all 3 products it produced very high concentration of dopamine in the brain, and which is continued for a longer period of time, is up to about 50 hours also it is present. For one case, you can see the dotted one, it is even at 50 hours, it still has a high concentration, which can provide the drug for longer period of time, which indicates that nasal delivery has taken the drug directly to the brain in a large quantity, maximum availability in the brain, and with a controlled release profile, that which indicates that the drug can be available for a long period of time. It is a different liposome we have prepared. Different liposomes are prepared, which is administered. You can see the different liposomes have different profile, which indicates that based on the liposome, your maximum concentration as well as duration of availability is different. So which is an important understanding that different phospholipids can produce different characters of the final formulation, depending on your need. One case, it is giving higher concentration, but it is declining quickly within 50 hours. It is coming to very low. Whereas other case, that Lipova E120, you can see the concentration is slightly lower than the other one, but it is remaining in high concentration till 50 hours, so which can provide the drug for a longer period of time. The same drug, when it is given by the same liposome we administered by intravenous route, in the right side profile, you can see that again, when administered intravenous, both solution and one of the liposome, you can see the drug reaching the brain is a very, very low from the solution, which is actual the picture for dopamine. But liposome has still gone very high quantity to the brain, which another information that if liposomal delivery is dopamine given, still through blood-brain barrier, more drug can reach the brain. If you see the pharmacokinetic parameters, you can see here that in case of pure drug by nasal route, the AUC is only 0.416, whereas three different liposome, it is 49, 156, or 77. The lipo by E120 is producing maximum AUC, where Cmax is 0.331 for the pure drug solution, whereas for liposomes, it is 2.37, 2.53, 2.74, which is maximum for the mixed phospholipids. Though for lipo by U120, it is 2.53, but area under the duration was much, much more. You can see the MRT for the pure drug solution, it is 1.33 hours, whereas for lipo by U120 is 55 hours, with compared to Lesiba S70, which is 17 hours, and the mixture case, combination of phospholipids, it is 22 hours. So which indicates that probably Lipova can be giving you the longer duration of action it is given. Similarly, summary of the pharmacokinetic of the intravenous administration of the liposome and the pure drug, you can see pure drug, nothing has gone to the brain with AUC 0.034, Cmax 0.024 microgram per gram of brain, whereas the phospholipids or liposome cases, it is still 10.62 AUC and 1.07, which indicates that even liposome can be administered, probably particularly this liposome, to get certain amount of drug to the brain, much more quantity. That is also another finding is there. Then the question comes, I mean about side effects, etc. What is the surety that from nasal route drug doesn't go to circulation also? So we studied the plasma concentration when that formulation administered by nasal route, and you can find it here. When the drug pure solution is given, a good amount is coming to the dotted line is the pure drug. A good amount is coming to the plasma or circulation blood. But for liposomal formulation, nothing much is coming. You can see more or less it is an x-axis. So not at all drugs are coming to the blood, which indicates that if it is administered by nasal route, the drug will not be coming to the circulation. So side effects cannot be there if the drug is having a side effects. On this basis, we worked on another drug which has been taken to the market as a nasal spray. So that is the important finding we can get, and you can understand by this process, many side effects and dose, etc., can be really benefited. the cost of the patient, for the patient treatment will be lowered, and patient will get the better happiness for that way, because no side effects, not frequent administration. So because administration also can be reduced. I will not take much time. I will have another drug case study, olanzapine. I am directly going to the again pharmacodynamic study. Formulation is not here. I am not discussing it as there, but because it is only 25 minutes time, I am not going to the formulation. Again, we prepared olanzapine liposome as well as solid lipid nanoparticles. You can see based on polymer, PCL polymer nanoparticle, as well as the solid lipid nanoparticle also. Here you can see again that as we studied in animal model, in the plasma drug concentration profile on the left side, you can see for pure drug, it is declining to the zero within 20 hours or less than 20 hours, you can say about 16 hours. Whereas for nanoparticles cases, both PCL nanoparticles and glycerin-based solid lipid nanoparticles, the concentration is much, much higher for the same dose administered to the animal by IV route. You can see, which indicates the drug remaining in the blood at higher concentration for longer period of time, because it is available till 40 hours. And if you see the pharmacodynamic parameters also, AUC for the solution is 7987, whereas the PCL case, it is much higher. But obviously, maximum is solid lipid nanoparticle case, it is 15,000, which is double than the olanzapine solution. Cmax also you can see 2290 nanogram per ml, whereas in case of solid lipid nanoparticles, it is about 1500, and for PCL-based nanoparticle, 1245. MRT, 3.66 hours for the pure solution, but PCL case nanoparticle is 8 hours, and for solid lipid nanoparticle is 10 hours, which indicates that when nanoparticles are given again, the drug administration to the brain and to the plasma is more remaining longer period of time. And if it is remaining longer period with higher concentration, you can understand the drug administration of permeability to the brain is also going to be more. Is it not the question of only statement? It is I'm presenting the data also. We studied also the drug amount per unit time or to the brain also. If you see the next slide again, left side diagram is the brain, where you can see dotted line, which is the pure drug. To the brain, it is a much, much lesser amount. As you see, the drug concentration nanogram per gram of brain is about 2000 plus, whereas other two nanoparticle product, it is a more than 4000, and in case of solid lipid nanoparticle, it is about 10,000 nanogram per gram of the brain. You can understand 5 times concentration higher is achieved in case of nanoparticle, particularly solid lipid nanoparticle. And if you see the duration, again you can see that in case of pure drug, it is declining about 20 hours in the brain, whereas for solid lipid nanoparticle, it is still available in high concentration, 46 hours, 45 plus hours, I can say. So that means your duration of action is going to be more, as probably it is giving a control release. And really on in vitro dissolution study, we found the nanoparticle product is providing the longer duration of release. That means achieved control release, that one. Now, it is a perfectly fine. The selectivity is good, you are saying it. But what about the toxicity? Can it also reduce the distribution to the other part of the? Yes, it is a very logically possible and happening, because try to understand, if same dose is giving higher plasma concentration in case of nanoparticle, that means less drug is going to the other part of the body. If brain is having more amount, logically, definitely than other part of the body, the distribution is going to be less. We did it with several organs. I'm presenting it with kidney only. It's due to lack of time. I'm presenting it with kidney. You can see here that again, pure drug case, the amount of drug in kidney is found to be much, much higher, more than 11,000 nanogram per gram. Whereas for nanoparticulate preparation, both PCL and solid lipid nanoparticles, it is much, much lower, where it is about 6,000 only or 4,000, 5,000. So which indicates that less amount of drug is going to the other part, in this case kidney, but we found in other organs also, which I am not presenting. So basically here we found it that when nanoparticles are administered, drug is remaining in the plasma for a longer period of time at a high concentration, which helps for better distribution to the brain, which we found it in the nanoparticle, more amount of drug is going to the brain. And the next point is also we found it that in case of compared to pure drug, the more drug is going to the brain, but lesser amount is going to kidney or other organ, which is important part, which I say, when you can make, then please you can try to understand that for a existing drug, if have a problem of non-selectivity, both binding to the receptor or the toxicity as well as non-selective distribution. Both are problem. Please keep in mind anti-cancer drug is a non-selective to the cells also because it kills the normal cells also. That is the reason patient suffers many anti-cancer drugs. So that if less amount is growing, that means it will not be able to produce that much of toxicity. That is the non-selectivity is also going to be less toxicity for that drug if we do novel delivery system properly designed. But important point to keep in mind, also it is indicating that what nanoparticle you are preparing, what phospholipids or what polymer using for the nanoparticle are also important. Many of our study with other anti-cancer drugs, which I'm not presenting here, we found it that for different drugs, different polymer or different phospholipid system helps a better delivery or a specific site. So it is possible that sometime changing the polymer, changing the phospholipid, or making a combination of phospholipids, a selective distribution is possible. So this indicates that it is possible to achieve better therapeutic efficacy, more selective distribution with less cost and better patient compliance by some novel delivery system. What inference we can bring it here that this study or result proved that the nanoparticle system can deliver drugs selectively, leading to lower dose. We can decrease the dose, cost will be reduced, low or no side effects, and with better therapeutic efficacy and better patient compliance. I think it is a very, very important approach and it can be achieved, which keep in mind also quicker to go to the market, quicker go take into the market because the drug is existing drug, which is already approved drug. I'll not take much time. I think my 25 minutes, because I'm very particular about the timing. I make my presentation according to the schedule, which is 25 minutes. I'll stop here because there may be some question or something. I must acknowledge funding agencies. Oh, sorry, ING is missing here. Industry and government agencies, because I have many industry sponsored projects where I deliver the product to the company. Obviously, my doctoral and graduate student who worked with me, BITS Pilani authorities who permitted me to work, and still I am continuing there, and my colleagues. I would like to one correction to the introducer who mentioned, I just recently retired from BITS Pilani. Yes, 8 years I was in Dubai. When I accepted this invitation, I was in Dubai. Then I became the Vice Chancellor of BITS Pilani Hall, so I moved to Pilani. And just last month, October, I only retired from BITS Pilani. Obviously, BITS Pilani, I have to show that 4 campuses. I worked in Pilani 28 years, total 35 years. About 8 years I worked in Dubai. Obviously, as Director of Research for whole BITS Pilani sometime 2010 to 14, I also worked with Goa and Hyderabad campus as I was driving the research agenda for the whole university in all 4 campuses. So thank you very much. This is the Saraswati Temple, Goddess of Saraswati Education, which is in Pilani campus. I got the blessings for 35 years. I worked for BITS Pilani. Thank you very much.
Thank you so much, Dr. Ranendra Saha. That was an amazing keynote presentation, and we are really honored to have you. It's a real privilege to have you at the 9th World World Conference on Pharmaceutical Science and Drug Manufacturing. Thank you so much, sir, that despite your busy schedule, you joined us, and we apologize for all the delay and the technical glitches that has happened today. But thank you so much, sir, for the amazing presentation that you have given us. Any questions for our speaker?
Yes, sir.
Prof. Shaha, good morning, good evening, Prof. Shaha. This is Dr. Lokesh from Malaysia. I would like to ask you one question. Actually, you choose the candidate dopamine. Basically, dopamine is a polar compound, and peripheral distribution of dopamine is very high, and centrally it is very poor because of the blood-brain barrier. It will not recognize by the genetic code. That's why the traditional practice, current practice is that the L-DOPA will be given to the patients, though it will be crossed easily blood-brain barrier because it's an amino acid. It will be recognized by the blood-brain barrier, and then it will be converted by the dopamine in the brain, and it binds to the receptors of the dopamine. So now in your delivery, you are using aerosol as a dopamine.
Not aerosol. I never said aerosol. This is a nasal spray only, not aerosol product.
Okay, nasal spray, whatever it may be, nasal spray or anything. So
no aerosol, no liquid propellant or nothing is there. Simply dispersion which is sprayed inside the nozzle.
Okay, dispersion in the nozzle. Okay, nasal delivery. Okay, nasal delivery. I will talk in this way. So how it crossed the blood-brain barrier, sir?
No blood-brain barrier. Oh, Vacha, I forgot to show you that one. Actually, when I giving to the inside, I have a picture. Unfortunately, to reduce the time, there is a place where you apply, it directly goes to the brain. It doesn't go to the blood-brain barrier. And that's why I showed that no drug is coming to the brain, blood at all.
No, but in your preparation, the dopamine is bind to any lipophilic.
It was the phospholipids I've used it.
Phospholipids again.
Phospholipids I've used the liposome.
Phospholipids are recognized by the brain receptors, the brain barrier, I know, but they are still polar, I mean, how
but it is directly going to the brain. It is not going to the blood-brain barrier at all. I can show you one picture.
Kindly, because I'm very curious to know, actually.
No, I can personally write to you. I can, if you send me mail, I can personally share with you. Okay, that is a mistake. I should have shown the picture. I thought people are aware. There is a nozzle, there is a specific place is there. If you administer, it goes. If you remember, I'm very simple way I'm telling you. Sorry, don't take it otherwise. People are taking cocaine, etc., the drugs for addiction through the nozzle. It directly goes to the brain. No need of blood-brain barrier at all.
Because cocaine structurally they are different actually, because dopamine is highly polar and very quickly metabolized peripherally.
But it is a liposome which is going actually there. Inside the liposome, the polar drug is hidden there.
Sure. I'm interested to know that. If you have that slide, it would be great to know. But
sure, I can show you that.
Olanzapine, I can understand because it's a highly lipophilic drug.
Actually, I tell you, our target was a separate drug. It is a tapentadol. But I first worked with dopamine because it is also a challenging. And then I worked with the tapentadol, similarly, which is taken to the market.
Okay.
It was patented and taken to the market.
What were the side effects you observed in the process? PK?
So because very less amount is coming to the dopamine case, I showed you, it is a very
dose is reduced because of nano delivery. Okay,
yes, it is a nano delivery directly going to the brain.
Thank you, bro. Thank
you.
Hello, sir. This is Pranav Raghavendra from PSG College of Pharmacy.
Yeah.
Sir,
I visited your college long time back to deliver a lecture.
Yeah. Thank you, sir. Actually, you told that there will be minimal side effect when compared to the conventional dosage form and a plasma route for antipsychotic drugs in class. But whether you have a report study for epileptic discharge or seizure activity upon targeting delivery to brain?
No, actually, I tell you, this is, particularly keep in mind, dopamine is a very well-known, olanzapine is a well-known drugs. Okay, yes. So there was no report of such things. That's why it was not a problem for that. We did not study it, but we have, that's why I'm telling that existing drug, which is otherwise very well-known, well-established, only side effects can be avoided. Because more drug is going to the target organ. Yes. It is possible tomorrow for lungs, it is possible tomorrow for the heart, kidney, any places, liver.
Yes. Thank you.
Thank you.
Thank you, sir, for the spontaneous reply and giving a lot of inputs to the questions. Anybody else have any questions? Okay. So then we'll move on to the next session. Thank you so much, sir, for joining once again. Thanks a lot.
Thank you. Thank you very much.
Yes. So next we'll be moving on to the technical session. So actually we have already completed 2 of the technical sessions. So before that, I would like to welcome our session chairs of the day. That is Dr. Jayavardhan Patil, Dean from the Department of Pharmacy, Sankal Chand Patel University from India, who will be evaluating the presenters at the physical venue at Dubai. And we have Professor Dr. Nahla Ismail, the counselor member from Malaysian Academy of Pharmacy from Malaysia, who has joined us here on Zoom and who will be evaluating the presentations. Good evening, ma'am. Dr. Nahla.
Hello, good evening.
Good evening, how are you?
I'm fine, thank you.
Yes, ma'am. So 2 of the presentations were already presented at the physical venue. That was by Dr. Inam and Mr. Sajid Majid. The next presenter who will be available to give the presentation from the live venue is Sumaya Aib from the University of Lahore from Pakistan, who has joined us at the venue at Dubai. And she will be discussing about the pH-sensitive compound liposomes of mesalazine and curcumin prompt instantaneous relief to guinea pigs, models of ulcerative colitis, which is an investigating drug synergy along colon targeting approach. So requesting the technical team to connect from the live venue for Sumaya Aib's presentation. Yes, welcome Sumaya Aib.
Thank you. Assalamu alaikum and good afternoon, everyone. My name is Sumaya Aib. I am an Algerian student of last year in pharmaceutics, MPhil pharmaceutics. I hope everyone is quite refreshed after the break now. Well, the topic of my research is pH-sensitive compound liposomes of curcumin and mesalazine, which has prompted an instantaneous relief in guinea pig models of ulcerative colitis. Here in my research, I am investigating drug synergy along colon targeting approach. So the reason I have invested my research in inflammatory bowel disease is because inflammatory bowel disease is a lifelong disease that may not be very fatal, but it could cause a lot of complications that itself, it could cause fatality in the future, such as colon cancer and so on. So because it is a lifelong disease, the patients of inflammatory bowel disease, they can go through a lot of discomfort throughout their life, and their daily activities could be disturbed. And the current management of inflammatory bowel disease is still poor, and it does not provide an efficient treatment for the disease itself. So we need efficient treatment strategies and advanced pharmaceutical techniques in order to provide efficacious treatment for these patients. Next slide. Next slide, please. Okay, so the previous one. Okay, so I would like to give a brief immediate note on inflammatory bowel disease. So it is an autoimmune recurrent inflammation of the gastrointestinal tract. So basically based on the extent of inflammation and the location of the inflammatory regions throughout the digestive tract, it is categorized into 2 forms. One is the Crohn's disease that you can see on the right. You can see a lot of areas of inflammation throughout the gastrointestinal tract. And the other one is on the left, ulcerative colitis, which only happens in the last section of the gastrointestinal tract, which is the colon. So because ulcerative colitis is the most common one and is the one that is responsible for colon cancer, I invested my research in this form. So yes, next slide. So the rationale of my study is, as I have already mentioned, the current management of inflammatory bowel disease relies on non-targeted therapies. That means that the conventional treatments that we have for ulcerative colitis, these are systemically absorbed. And we all know that the drugs that are systemically absorbed, they do not reach to the target site. Instead, they are absorbed, and a lot of the drug could be lost before reaching to the target site. Also, we know that drugs which are systemically absorbed, they may go through the systemic circulation to other body parts and could cause serious side effects. And of course, if the drug does not reach to the target site, we know that the efficiency or efficacy is not increased. There will be no proper treatment, and we have to increase the dose, which of course may cause again side effects. So in my study, this is a novel therapy for inflammatory bowel disease. So the hypothesis of my study is basically I have formulated a system that targets the colon specifically. For this, I have used the pH-sensitive approach, which means that the drug, based on the pH, it will directly be delivered to the colon based on the pH of the colon. So when it enters, it does not be systemically absorbed in the stomach. Instead, it goes directly to the colon, and the drug is released there and can give better efficacy. So there will be more amount of drug in the colon, and the administration dose that we give in conventional non-target therapies, we can reduce them easily because we don't need a lot of dose to provide that much of efficacy. Again, it's not systemically absorbed, so there will be no side effects. So again, in my last hypothesis for this study was that if I include two anti-inflammatory drugs, it will give me a synergistic effect. We know that synergistic effect is an effect of two drugs that have the same effect, and so we could reduce the doses of the two drugs and we could have better efficacy. Next slide. So this is the system that I have designed. It includes two anti-inflammatory drugs. Mesalazine is a drug that is currently the best drug out there in the market for inflammatory bowel disease. However, we know that it's conventional, it's non-targeted, so it has side effects and so on. The other drug I have chosen is curcumin. I thought let's choose a natural source anti-inflammatory. So the system, as we can see, since mesalazine is hydrophilic in nature, we have entrapped it inside the hydrophilic core of the liposome. So this is the nanotechnology system that I have used is a liposome. So the reason I have chosen a liposome here is because liposomes are the nanotechnology that can entrap two drugs inside. So mesalazine is entrapped inside, and curcumin, since it's hydrophobic, I have embedded it in the lipid bilayer. And then I have covered the system with Udrajet coated. I have chosen Udrajet S100 because it is a pH-sensitive polymer, which is going to deliver this system directly to the colon. So the drug reaches immediately to the colon, and once this coating is broken inside the colon based on the pH, we can have the drugs released there. Next slide. So this is a graphical presentation of the entire experiment. So in order to check to validate my hypotheses and to check whether this treatment is going to work for such patients, I have checked it on guinea pig models of ulcerative colitis. For that, I have to induce the ulcerative colitis first. So I have chosen the method of acetic acids. It was intrarectally administered in the guinea pigs, and then I have induced about mild to moderate ulcerative colitis because that's the most common form of ulcerative colitis. So I had to examine that. And then we had three treatments. The first treatment is our optimized treatment that we need to validate, which is a coated pH-sensitive curcumin, mesalazine, liposomes. The other two treatments that we needed for comparison is a normal solution of mesalazine alone, and then other liposomes that are not coated with pH-sensitive polymer. So we gave an oral treatment, and then as you can see, that is my system. So as we can see on the right side, that if I'm giving the my optimized formulation, the coated liposomes, the coating will be broken about in the pH of intestine to the colon. We can see the drug is delivered at the colon pH 7 to 7.4. However, if we're giving plain drug solution, as it's stomachally absorbed, it does not depend on the pH. So as the drug enters the stomach, the drug is immediately released throughout the digestive tract, and only minimal amount of drug is available for colon targeting. Next slide. Okay, so first of all, here I have done in vitro and in vivo studies. For in vitro studies, I have done it to check how much stable is my formulation and to check the efficiency of the system as a whole. So first, we have characterized it physicochemically. We have checked its physical size, zeta potential, and homogeneity to see what is the homogeneity and what is the dispersion efficiency of the system, and then also the entrapment efficiency. We wanted to know how much the drug is efficiently entrapped within the system. And then we have optimized our formulation. Of course, there were a lot of factors, the chemicals that we have used to design this formulation, and then and the amount of those chemicals, it had a lot of variations. And so at the end, the optimized formulation that we have chosen and that has the best qualities was the one that has a physical size of 212 nanometer, which is a good size, because particles of 200 to 500 nanometers, they get endocytosed by the macrophages, which are available in the colon. There's inflammation. So once the macrophages engulf these particles, it is better because the drug is retained there in the inflammation. So the efficacy would be increased. The zeta potential is negative 29, which indicates that the coating is stable there, and that the drug is going to be directly targeted to the colon. The PDI is the polydispersity index, which is 0.14, which indicates that our formulation was quite homogeneous, and then and that the dispersion was quite well. The entrapment efficiency that of the optimized formulation was 78 for mesalazine and then 84.7% for curcumin, which is again indicated as a sufficiently good entrapment for drugs. Then we have checked the pH stability of the of the formulation, excuse me. This is because we wanted to see that if the drug is physiologically gone inside the body, what is the effect of pH? How is the drug going to perform? So we have first checked it in vitro. So we have checked it for pH 1.2 because that represents the pH of the stomach, and then again for pH 7.4 because that represents the pH of the colon. So for the uncoated drugs that did not have the coating of pH sensitivity, we saw that the vesicle size were quite increased. And this represents that, of course, the pH has increased. And of course, we know we don't want the vesicle size to be increased or reduced. So there were physiological alteration in the vesicle size. And again, the PDI was reduced from increased from 0.14 to 0.29 again. So the homogeneity and heterogeneity of the system was disturbed. The zeta potential was reduced, which means that there's no coating. The drug is not delivered to the colon. Instead, it's delivered to the stomach. And then at pH 7.4, which represents the colonic pH, we have checked it for the colon, our optimized formulation. The vesicle size is 212 to 201. So it reduced. So this shows that at the colon, the vesicle size had reduced because the drugs have been released in the colon. So once we have checked it in the colon because of the pH, this indicates that the vesicle size has reduced because the drugs have been released in this colonic environment. And then the zeta potential again reduced from -29 to -25, again indicates that the coating has been broken only on the colonic pH. Then we have checked the storage stability because the effect of temperature, we wanted to see that whether how well our formulation performs at temperature. So for coding liposomes, we know that the storage stability is 4 Celsius for the liposomes. So at 4, there were no alterations in the storage stability. However, at 25, we have checked it for 3 months. Now, throughout the days, especially at the last month, we have seen alterations in the stability. The vesicle size has changed, the PDI as well has changed, and the entrapment efficiency reduced from 78 to 58, which means that the drug has released from the system when it was stored at pH 7 at our storage temperature, which is 25. Next slide, please. Okay, so that is the last in vitro study where we have tested. So it is called in vitro drug release pattern. So on this, it gives us an overview of what is the behavior of the drug as soon as it enters the body. So we have checked, we want to see here that how does our drug or our formulation behaves as soon as it enters into the body. Okay, so we have checked it at pH 1.2 and pH 7.4. Again, the pH 1.2, it represents the stomach pH, and pH 7.4 is representing the colonic pH. So as you can see, we have 4 formulations here. The circle one is mesalazine and curcumin from the uncoated liposomes, and then the triangle ones, it's mesalazine curcumin from the coated liposomes, which are our formulation. We can see if at pH 1.2, the drugs which were not, the system which is not coated, it was all released in the stomach at pH 1.2 for 2 hours. As we can see, the circle ones, immediately they are at about 82% of the drug was released in the stomach. However, for the coated drugs, our optimized formulation, the drug was minimally released at the pH 1.2, which is stomach, and then gradually it started releasing as the pH was changed into 7.4, which is the colonic pH. So here we have validated that yes, our optimized formulation is actually released based on the colonic pHs and in a sustained release manner, which can even increase or prolong the duration of the efficacy of our formulation. Next slide. Now we come into the in vivo studies, which are basically we're checking our formulation in animal models of ulcerative colitis. So here we first are evaluating our therapeutic efficacy of the formulation. We have done a macroscopic assessment of the colonic damage and a microscopic assessment. So microscopic assessment, we see that how much colonic damage has is there and how much it's treated with our treatments. And then the microscopic, we see some histopathological alterations of the inflammation, and we can characterize that. Next slide. Next slide, please. Okay, so for macroscopic assessment, first we can see the left one is the untreated group. We can see a histological score, which is very high. This represents a high, the extent of inflammation is very high. And then for the next one is mesalazine solution. We have seen that the histological score have reduced, which means that the solution, which is uncoated, it has recovered the inflammation, but not as much as the other two treatments. The right one, again, it's a single mesalazine liposome. It is coated, but it has only single drug. So it has treated inflammation, but again, not to a certain level. And then the final treatment is mesalazine curcumin liposomes, which are coated and having two drugs in a synergistic effect. And so it is the best one that has treated and reduced the histological scare for a very good level. Next slide. So in a microscopic assessment, we have seen histopathological alteration. The first a slide here shows us the healthy control groups. We can see a normal architecture of the colon. There are no histological alterations. In the second slide, we can see the negative control group. This is the group that is diseased. We have given it the ulcerative colitis, but we have not treated. So we use it for comparison. So you can see that we have denoted that the mucosa of the colon, it is destructed. There are inflammatory cell aggregated there, so it indicates inflammation there. And then there's a slight degree of hemorrhage. The next slide is the group that was diseased and has been treated with a single plain mesalazine solution. We have seen that the inflammation has recovered a little bit, but there's still a damage in the epithelial lining of the colon. And then again, there's slight vessel congestion. So it indicates that the inflammation is still present there. And then in the other treatment down there, single mesalazine liposomes, again, the inflammation has recovered up to a very good level. However, we can still find a slight mucosal congestion there. And finally, our optimized formulation, we can see the architecture of the colon is very normal. Just if you compare to the healthy mucosa of the colon, it's similar to that. So that means that our optimized formulation, it has recovered inflammation up to a certain levels. And that means that our formulation, which releases the drug specifically at the colon, has worked very well. Next slide. The next in vivo study, it is called liposomes localization efficiency. Here we want to see that our optimized formulation, how well does it retain in the inflammatory colon. So we can see that here we have a 3 level of the gastrointestinal tract. We have checked it in the stomach, small intestine, and then colon. So first we have given, we've seen, we cannot see because of the banner there. So the first one was a single mesalazine solution. We have seen that still the drug was not retained. It was retained first in the stomach and then more in the intestine, and then the colon was not retained there. So the drug has no pH sensitivity. And then the other drug containing, which is a liposome but containing only one drug, which is mesalazine, again delivered in the stomach and then intestine and then in the colon, not too much because it's a single drug. And then at the end, our optimized formulation, which is pH sensitive and releases a drug in the colon. We have seen that it has released the drug in a very minimal amount in the stomach and then a minimal amount in the intestine, and then a high degree has been released in the colon, which validates that the drug, yes, it is pH sensitive and releases the drug immediately into the colon. Next slide.
The other test for in vivo, we have quantified the cytokines. Now, what are cytokines basically? These are pro-inflammatory biomarkers. So in inflammation, these biomarkers are increased. So these are a sign of that there is an inflammation. So first of all, we have chosen interleukin 6, interleukin 4, and tumor necrosis factor, and a TNF. So for interleukin 6, we can see that in the diseased group, these cytokines are increased, which means that the disease group has a lot of cytokines, which indicates inflammation. Now, in the others, which we can see 65% of interleukin 6, this represents the single mesalazine solution, which means that the cytokines expression has reduced and indicates that the inflammation has reduced up to a certain level. Again, for the other treatment of single mesalazine liposomes, we have seen that the inflammation again has reduced, but not up to not completely. Now, at the end, we have 16% of interleukin 6 expression, which is for our optimized formulation, and we have seen that the level of the cytokine interleukin 6 have reduced drastically, which means the inflammation has recovered. Similarly, for TNF alpha, these are the same results, and it validates that our formulation again has reduced the cytokine more in the optimized formulation. Next slide. Similarly, for INF, again, the same results we have seen that for our optimized formulation, the cytokines has reduced the best, and again indicates that the inflammation has recovered. Now, for interleukin 4 works opposite to these cytokines, especially in inflammatory bowel disease. So its level of inflammation decreases instead of increasing. So we can see that in the end, with our optimized formulation, the level has increased the most, and which again indicates that the inflammation has recovered. Next slide. Now, this is another in vivo study, which is biochemical antioxidant assay. Now, in inflammatory bowel disease, oxidative species produce, which causes oxidative stress. So for that, we need to calm it down. So these antioxidant, we have two antioxidant enzymes. What they do is that they act as a defensive mechanism against the oxidative stress that is present in inflammatory bowel disease. So we have chosen reduced glutathione assay and then glutathione sulfotransferase. So if the level is increased, we know that they are working efficiently, and the oxidative stress is decreasing inflammatory bowel disease. So as we can see in the left one, we have, this is a healthy control group, which shows it is slightly high here because it shows the body's natural defensive mechanism. And then as we move to the treatments, which are single mesalazine solution, the disease group, and then the single mesalazine liposome, we see that the level has gradually been increasing, but not to the same level that our optimized formulation has shown. As we can see, our optimized formulation of mesalazine and curcumin liposomes has shown the most the highest level of GSH, which indicates that the oxidative species have been defended, and then the oxidative stress have gone. And then similarly in the GST, again, we can see that our optimized formulation has the highest level, of course, compared to the natural body's defensive mechanism in the healthy group. We can leave that. But for the disease groups, our optimized formulation showed the highest level of GST, again indicating that the oxidative stress have reduced the best with this treatment. Next slide. Now, this is the final in vivo study, which is a biocompatibility test. So we want to know here that if we're taking our medicine, is it safe for our body or not? So for that, we have tested these two treatments. First, we have tested our coated mesalazine curcumin liposome, and then the other single mesalazine solution, which is not being delivered through a liposome, instead just a solution. So we want to see both, we want to compare the safety of both. So we have checked it on heart, on the kidney, on the liver, spleen, stomach, and small intestine. Now, there were no alterations, there were no changes we have seen in other organs except in the heart and in the kidney. Now, for the heart, the first square on the left shows a normal architecture of the cardiac muscle. This is the healthy group of the guinea pig. Next to it is the diseased group. We can see that there is inflammatory cells in the cardiac muscle and also slight congestion of the blood vessels. This may indicate that a slight myocarditis have occurred, which of course in mesalazine, if you're giving mesalazine in a non-targeted therapy as a solution, it is reported in the literature that it may cause a slight myocarditis or inflammation of the cardiac muscle, especially during the first days of treatment or following prolonged treatment course. And then the final slide shows our optimized formulation. It shows that there is no alterations in the cardiac muscle, which proves that our optimized formulation is safe for this organ. Next slide. The other changes that we have detected is in the kidney. The first slide again shows a normal renal structure and morphology. The next slide in the center shows the disease group. Now the disease group has, we can detect that the size of the glomerulus has increased slightly, and we can see slight cellular proliferation of inflammatory cells. So again, it may indicate actually renal inflammation. And again, it is renal manifestations in inflammatory bowel disease can occur. However, because of the harsh symptoms of inflammatory bowel disease, sometimes it overshadows the renal symptoms, and we may not know that there are renal symptoms. But however, if we're taking a solution form of treatment, we have proved that yes, it causes side effect in the kidney. And then the last slide, it shows our optimized formulation again has a very normal morphology of the kidney, and so it has no side effect on the kidney. Next slide. So the conclusion, we have first checked our formulation properties. We have seen that it has very good physiological characteristics, ideal physiological and storage stability, pH-dependent sustained release manner, colon-specific delivery, strong retention efficiency, enhanced synergistic effect because these are two anti-inflammatory drugs, and reduced therapeutic dose because we are giving two drugs and we know that it's going to direct the colon immediately, so we need to reduce the doses. We don't need a lot of drug. And then high antioxidant activity, we have proved that both drugs have antioxidant activity alongside anti-inflammatory, and there are no side effects. So we have proved that our mesalazine curcumin coated liposomes are very efficient since they are synergistically shows synergistic efficacy, and also we have seen that reducing the dose tenfold than the normal dose that we give inflammatory bowel disease can produce better efficacy even than the normal treatments that are out there in the market. So the hypotheses have been validated, and now we can validate that mesalazine curcumin liposomes is an efficient treatment or promising therapy that needs to be validated as the marketed product for the treatment of inflammatory bowel disease. Thank you, and thanks a lot for sharing your story. If there are any questions, you're welcome.
Thank you so much, Sumaya, for your presentation. Professor Nahla, any questions for Dr. Sumaya?
Okay, Dr. Sumaya, can you hear me?
Yes, yes, ma'am.
All right. A very good presentation. I think you covered quite most of the aspect for your study. Your study is looking at the synergistic effect of the CM and NZ. Okay, I may miss a part of the from the beginning because I couldn't hear. It wasn't audible on my side. But I'm just wonder the one that you use for control. Can you just explain?
Exactly which slide, ma'am?
Sorry.
Which slide or concept are you talking about so that I can
No,
explain?
I'm asking the one that you use for control. Can you just describe a little bit? On the control part. When you do the in vivo study for the control, is it just like a typical? I think you use mouse, maybe mice.
No, these are not mice. These are guinea pig models. So they're
You use guinea pig. Okay, so for the control group, the guinea pig is it normal?
They are very slim. Their weight is decreased, so they cannot bear it. So we have chosen male guinea pig models that can bear it. So it was a treatment, an experiment for 3 months, which we have checked throughout.
Okay. Yes. In your results, okay, you have shown the one that only MZ, and then MZ single liposome, and then MZ and curcumin.
Yes.
I'm just wondering the result for just curcumin. Do you have it?
No, ma'am, because we know that in inflammatory bowel disease, mesalazine is the best treatment that is in the market. But still, we have a lot of side effects, and it's unable to control the disease. Still, patients are going throughout their life. So what we did is that we brought a normal mesalazine solution that is being sold in the market. And then again, we focus on mesalazine as a therapeutic drug for inflammatory bowel disease. So we have delivered it through liposomes in a coated manner to check that if we're giving mesalazine, it's good, or maybe the synergistic effect by adding another inflammatory drug would be better. So we have compared single mesalazine liposomes as well as double drugs, which are mesalazine and curcumin. So our target basically to check mesalazine more compared to curcumin. Curcumin was just an adjacent adjuvant therapy to mesalazine to show that synergistic efficacy can work very well in inflammatory bowel disease.
Sorry, Dr. Sumaya, I can't really hear you because, you know, like in your result, I haven't seen anything with just only curcumin, because your study is looking at the synergistic effect. So I'm just wondering, you know, if it's just curcumin and the single liposome, yeah, I mean, before it is combined with the MZ.
Yes.
So I'm just wondering because it wasn't shown in the results. The result is just focusing on the MZ, and then MZ with the single, and then MZ with the curcumin. But I couldn't see anything that just curcumin first, you know, because your topic is looking at the synergistic effect. So I'm just wondering.
Ma'am, as I told you, because I did not check the efficacy of curcumin alone. Why? Because I'm focusing more in mesalazine.
Sorry to interrupt. Dr. Sumaya, your audio is low, so the session chair is not able to hear you well. Can you please hold the mic closer? I request the technical team to kindly provide another mic so that it's audible.
Can you hear me now?
No, little louder.
Now? Maybe now.
Ma'am, is it possible for you to follow her now? Yes, Dr. Sumaya, you may continue the explanation.
Can you hear me now?
Yes.
Okay, so the answer is that I think the doctor asking me that why didn't I check the efficacy of curcumin alone as I checked it for single mesalazine. That is because our main agent here is mesalazine, because it is currently the best treatment out there in the market for inflammatory bowel disease. So we're focusing more on that, but we know that with the treatment that are available in the market of mesalazine, we know that there are a lot of side effects, and still patients are not being treated. This is a lifelong disease, so we need a prolonged remission for this disease. So we have focused more on mesalazine, and then the hypothesis was that if I include another adjacent agent with it that can act as a good synergistic efficacy with mesalazine, we can see then how well the system would, the entire formulation was produced. So we have seen that if I'm giving mesalazine in a normal solution, no liposome, no colon targeting potential, it is giving me side effects, still no good efficacy, and the dose is increased. When we saw that we have given it through liposome, which are coated, of course, inflammation was treated very well. However, it was not as good as when I have added curcumin to this therapy, and the dose of both drugs have been lowered. So it gave me a synergistic efficacy, and the dose was reduced, and ultimately no side effects were there. I hope I was clear that mesalazine was the main agent here. However, curcumin was just an adjuvant therapy, so to produce synergistic efficacy.
Okay. Thank you, Dr. Sumaya. Dr. Nehla, do we have any other question?
No, but I'm not sure about my counterpart.
Sorry, doctor, I'm not sure if I have quite understood question. But if you're asking me why I have not checked curcumin individually, is that so?
Yeah, I mean, that was the question that I asked at very first time.
If that's the question, actually, ma'am, in our research, curcumin has been checked for inflammatory bowel disease alone, and it has produced good efficacy. We know that curcumin is a potential anti-inflammatory agent in inflammatory bowel disease. However, our main hypothesis here said that if we included mesalazine along with curcumin, both which are potential anti-inflammatory drugs, is it going to produce better efficacy, reduce the dose, required dose, reduce the side effects. So yes, we have proved that. As long as testing mesalazine alone, it was only to validate our hypothesis that the marketed products out there for mesalazine, they are all solution, conventional non-targeted therapies. So if we're giving mesalazine in a targeted, colon-targeted therapy, is it producing better results or not? Yes, it is producing. We have validated that as well. So true hypothesis here is that if we're giving mesalazine in a colon targeting therapy, yes, it produces better efficacy. And then if I add curcumin to it or any other drug that is anti-inflammatory in nature, would the synergistic efficacy give me better results again? Yes, it did. That's what we validated for the two hypotheses.
Okay, thank you. Thank you so much. We have another session chair live at the venue, Dr. Jayavardhan Patil. So if you don't have any questions, shall we move forward?
Yeah, from my side, yes.
Okay, then. Thank you. So I would read out some presenters. If any of the presenters available at the venue, please, you may present. So as per the agenda, we have next Colombo Miriam, Senka Vidovic. Okay, and another presenter for the day was V. Purna Pushkala, who is unable to join today. So session chairs, the presentations for the day is over, and the presenters who had given presentations today is Dr. Inam Sameh, Sajid Majid, and Sumaya Aib from the present technical sessions. So with this, we come to the end of day 1 of the 9th World Conference on Pharmaceutical Science and Drug Manufacturing at Dubai. So next, before we wind up, we'll have the felicitation of the keynote speaker. So we have here Dr. Mohammed Haitham Aid, who have joined here, and I would like to invite Mr. Siddharth Charger, the Managing Director of Technorate Group, to kindly felicitate our keynote speaker. Welcome, sir. Congratulations and thank you to our keynote speaker, Dr. Mohammed Haitham Aib, who have joined us here. Thank you so much, sir. Next, I would request Mr. Siddharth Charger to felicitate our young scientist for the year nominated in the year 2021 for the 9th World Conference on Pharmaceutical Science and Drug Manufacturing is Dr. Jayavardhan Patil. Congratulations, sir. Thank you, sir. Thank you, Sid, sir. With this, we come to end of day 1. The remaining presentations will be continued on day 2, and the best presentation, oral presentation, and best poster presentation and best research paper awards will be given tomorrow. And with this, we come to the end of day 1. Thank you, everyone who have joined us live at the venue at Dubai and who have joined us at Zoom platform. And those who are watching us live on our YouTube channel. Thank you so much, everyone. Have a great evening.
Good evening, gentlemen. So it was nice a day today, the 1st December 2021, after a long time, 2 years back. We are here physically in Dubai. So I would like to thank and congratulate to the organizer, the Association of the Pharmaceutical Research. Really nice arrangement here in the hotel venue. And it was a nice talk by the scientists of the global, and nice presentation from the scientists, young scientists, and the speaker, keynote speaker like my friend. So it was amazing experience for me also. So anytime chance for me, I would like to join again the such type of the conference. So thank you so much, and see you tomorrow again. Thank you.
Thank you, sir.
In fact, it was a really pleasure to be in person and not in front of a computer after all these 2 years, So I would like to thank the organizers for, putting together this nice gathering, very interesting talks from different participants from different countries about different topics related to life sciences and drug development and production. So I really enjoyed it, and I will looking forward for another one, hopefully. Thank you.
Thank you, sir. Have a great evening.
So hello people, it was my pleasure to be today presenting at this conference. I thought that the conference was very informative. Every presentation we have learned from it, especially from the keynote speakers. And we thank all the listeners that they have shed ears to us, and we hope to see you tomorrow as well for more presentations. Thank you.